All right. Well, good afternoon to those in the United States. Good morning to those that are in Japan, and good evening to those in Europe. This is now our poster session time for the ISSR&D conference, and it lasts from 9 until 5:20 or so U.S. New York time. So we've got about 5 hours of posters to go through with you guys, and it'll be an interesting experience. These will be 10-minute sessions that will include live Q&A with the poster authors. And as I tell you about the process, then we'll also run through the agenda. Each poster will first display the poster and then play the presenter pre-recorded narration. When the pre-recorded narration is over, the presenter will be on screen for the remainder of the 10-minute period to answer your questions. Now, those questions need to be submitted through the Q&A tool, and I will feed the presenter the questions for him or her to answer. So that means you really need to be quick on your fingers and type in the questions to the Q&A tool. The Q&A tool is a little icon down at the bottom of your screen that says Q&A. Type in there, scroll down to the bottom, and type in your question, and make sure you hit submit question. And we are going to be moving very rapidly through this. Please do not submit your questions on chat. We're only going to be accepting questions through the Q&A tool, which is too much to mechanize otherwise. So our very first of the day, and this is a real experience for us because we haven't done posters this way before, so it'll be fun. First of the day is microgravity and partial gravity fluid physics, an experimental and computational study of bubble formation and detachment. And the presenter is Paul Burke, PhD candidate at Texas A&M University in the Aerospace Engineering Department. So can we play his poster, please?
My name is Paul Burke, and I'm a PhD candidate at Texas A&M University. My co-authors are my advisor and Aerospace Human Systems Laboratory Director, Professor Bonnie J. Dunbar, and recent master's graduate, Daniel Varnum Lowry. I'm presenting our work, Microgravity and Partial Gravity Fluid Physics, an experimental and computational study of bubble formation and detachment. As the United States plans to return humans to the lunar surface by 2024, there is a need for fluid systems such as ISRU, life support, and cryogenic fuel systems which operate in lunar gravity. Figure 1 shows NASA's Artemis mission concept. To date, there are no empirically validated CFD models of two-phase fluid flows in partial gravity. AHSL's specific research is focused on the balance between buoyancy and surface tension forces on a gas bubble in a liquid during bubble detachment. In 1G, buoyancy forces dominate, while in microgravity, surface tension dominates. Partial gravity models must account for both buoyant and surface tension forces. Figure 2 shows simple force balance diagrams displaying the buoyant and surface tension forces. On the left is a bubble forming through an orifice in a wetting substrate, and on the right is a bubble forming on a non-wetting material. The amount of bubble spreading is dependent upon material surface energy. AHSL has developed a CFD model using OpenFOAM and the computing power of the Texas A&M HPRC supercomputer. The variables were gravitational level from 0G to 1G in 10th G increments, including lunar and Martian gravities. The other variable was base plate material. The first materials chosen were polycarbonate and quartz. To validate the CFD model, AHSL has constructed an experimental platform to image bubble detachment in Earth's gravitational environment. The experiment includes a dual-axis high-speed imaging system to measure the bubble volume. On the top right, figures 3A and 3B show typical experimental and CFD sequences of bubble formation and detachment on polycarbonate in Earth gravity. The CFD model was used to measure the bubble detachment volume at each gravitational level on quartz and polycarbonate. Figure 4 shows the curves of bubble detachment volume on the y-axis versus gravitational level on the x-axis. As expected, surface tension dominates over buoyancy in microgravity. For a bubble not in contact with a surface, the gas volume and buoyancy are predicted by the bond number. which scales linearly with gravity. However, the bond number does not consider an interfacial tension force when the gas bubble is in contact with a solid surface in a liquid. The CFD data indicate that as gravity decreases, bubble detachment volume increases non-linearly according to a power law equation. The constant A and the exponential component B are decreased as the material's surface energy increases. Upon experimental validation in partial gravity, the results will impact heat and mass transfer models and the material selection in reduced gravity two-phase fluid systems. Future AHSL testing will include additional materials such as stainless steel and aluminum. In summary, in an effort to support the national research interests in lunar habitation, the Aerospace Human Systems Laboratory is developing a CFD model of bubble formation and detachment across variable gravity levels, particularly the partial gravity levels of the Moon and Mars. AHSL has found a nonlinear relationship between bubble detachment volume and gravitational acceleration, which will impact the modeling of two-phase fluid flows and material selection for fluid systems on the lunar or Martian surfaces.
All right, very good. Thank you, Paul, for your presentation there. And at this moment, we do not have any audience Q&A. So what we'll do is, until we get some Q&As, we'll open the floor and let you explain anything additional you want to explain for your poster.
Yes, thank you. So thank you all for tuning in and viewing my poster. I'd like to thank my co-authors, Professor Bonnie Dunbar, Director of our Aerospace Human Systems Laboratory, and my advisor, and recent master's graduate from the lab, Daniel Varnum Lowry. So this is ongoing work. We are continuing to refine some of the CFD models, and now our next phase is really concentrating on varying the material selection in the CFD models to obtain models that scale across multiple surface energies, varying from quartz glass to stainless steel, aluminum, and polycarbonate, as I discussed in the poster. We will continue to run those CFD models and plot each curve of the materials across these variable gravity levels from 0G all the way up to 1G, and continue to obtain these power law equations that scale. We see a very nonlinear behavior and sharp jump in the bubble detachment volume around lunar gravity, which, as the US looks to go to the moon within the next few years, by 2024, this will be very important for material selection and heat mass transfer models on ISRU design, Eclipse design, and things like that. I think we have a Q&A.
Yes, we have a question, Paul, and the question is, do you have a flight experiment?
Yes, so we do not currently, we do not have a manifested flight experiment right now, but we are designing, in the preliminary stages of designing a few different flight experiments, both for the ISS on an artificial gravity platform, which would induce artificial partial gravity, as well as possible lunar landers using the CLIPS mission. Dr. Dunbar is more the contact for programmatic and funding questions for our flight experiments and future work. So our email addresses are in the bottom, and I think we could detail that a bit more.
Okay, Paul, and does this apply to nanobubbles?
Nanobubbles, yes. So any bubbles that are in contact with a solid surface, so any two-phase flows, water or liquids and gases that come in contact with solid materials, it would apply. Right now, we are working with an orifice of 0.4 radius, 0.4 millimeter radius. So our bubbles are fairly small. In 1G, they're around 10 to 20 microliters, and I believe it could go lower. We chose that orifice due to some past literature, and that's a typical orifice size for these types of experiments, but we believe the models could scale down to nanobubbles as well.
Okay, thank you very much, Paul. This is an interesting poster. I actually read it and understood a little bit about it, but not much. So thank you, and thanks, Bonnie, for supporting you in this, and wish you well as you go forward.
Great, thank you all.
Our next poster is Carcinogenesis in Space by Souchelnytskyi. And I really bungled that up. So help us with your name, Cheryl, when we get to you. She is at Qatar University. So play their poster, please.
Hi everybody, my name is Serhiy and I will tell you a few words about this project. When you look on this poster, you look on the project aiming to improve treatment of cancer with use of space technologies. Today people die from cancer. This shows that all our great science has not delivered yet the solution. We have great improvements, but as long as people die, it's not enough. We have to look for alternative ways to study this disease and look for better treatments. Carcinogenesis is a process driving transformation of normal cells into malignant. This process includes changes of cell physiology, proliferation, migration, death, differentiation. Carcinogenesis involves cancer driving cancer-specific mechanisms, but also mechanisms engaged in normal cells. We want to separate, we want to focus only on cancer-specific mechanisms, because that would allow us to control them, and that would lead to better treatment. To find cancer-specific mechanisms, we have to find new ways of interrogation. Spaceflight offers a possibility to study carcinogenesis under conditions not familiar to cancer cells. This non-familiar environment will force cells to maintain cancer-specific mechanisms. Cancer phenotype would be, in this case, preserved at all three types of conditions we want to study: spaceflight, weightlessness, 1G, surface of the Earth, and 10G. We aim to detect these cancer mechanisms and develop drugs that would eliminate cancerous cells. In the space, cells would be exposed to weightlessness, slight decrease of gravity force. Impact of gravity would be different from the surface of the Earth due to the Moon and the Sun, and that may have an impact on cellular biochemistry. So what we want to do? We will let cancer and normal cells to grow in the space and compare them to the cells at 1G and 10G. Then we will analyze their carcinogenic properties and identify regulatory mechanisms engaged in the cells. Extraction of these mechanisms will include a systemic analysis of regulatory networks specific for weightlessness, space versus 1G versus 10G, related to carcinogenesis, and we will compare to known cancer hallmarks, diagnostic signatures, and treatment markers, companion diagnostics markers. So far, we completed 7 simulation runs with incubation of cells for 4 to 6 weeks and mimicking conditions of the experiment. Microtumor formation, monitoring of cellular proliferation, morphology, and death, and the proteome maps are the current results. Markers of clinically used signatures are included in the analysis. Functional molecular diagnostics, FMDX, is an example of clinical applications we want to do. And this is a way how we want to introduce our findings into clinics. This shows that the technology is ready. We have tested the workflow and now preparing for the space flight. For the flight, we designed it and produced a biomodel in a format of 2 ice cubes. The flight is scheduled for summer 2021. The expected outputs of practical and exploratory impacts. Practical impact will be by diagnostic signatures and novel ways to develop treatments. Exploratory, this is to study how space affects carcinogenesis, as physical forces up there are different. Differences in weight, mass, gravity, electromagnetic waves, particles, and even time. We will look on proteomes, clinically relevant markers. The cells will also be prepared for studying of genes, transcripts, and we will look on secretomes. We are open for collaboration, as the cells will be back to the Earth and expanded. Response to drugs, tumorigenesis in animals, different arrays, sequencing, and metabolomics are examples of what could be done in addition. Cancer is a complex disease, and together we have a chance to win the fight. In summary, our aim is to explore how carcinogenesis is affected in the space and use the results in clinics. Thank you for your interest. With best regards from me and my team, have a great conference. Bye. Bye.
All right, so thank you for the poster, Serhiy. We do have one question, and that is, over which duration will a 10G stimulus be applied? Okay, you need to turn on your mic and your camera, please. There you are. Okay,
Do you hear me?
The question was, over which duration will a 10G stimulus be applied?
For flight, I would prefer for a long time, but limitations of scheduled flight is 4 weeks.
Okay.
So answer is 4 weeks.
All right. And we don't have any other questions at this point. So if there's more you'd like to talk about on your poster over the next 4 minutes, you can do that, and I will interrupt you if we need to bring in a question.
I mean, maybe I have to start that I'm actually a guy, gentleman, but that happens with my name, Serhiy. Then about poster, this is actually a project which has been going on since 2018. First was a lot of preparations in the standard labs. Then we looked whether we can we actually get something new flying up there compared to what we can do down here. And it looks like, yeah, we actually can, because there are some indications that there are differences in biochemical reactions in the space conditions compared to Earth, and these differences cannot be explained by radiation, magnetic waves, particles. Something else is happening there. Probably link it to gravity, maybe link it to something else, we simply don't know. But point is, this is another additional trigger to cells to expose what is cancer specific. Just to give you in numbers, today when we look for patients, because key activity is actually with patients. I have more than 50 patients who used this type of technology already. And when we looked on patients, we see really thousands of proteins and genes changing their activities. And I know for sure that majority of these genes and proteins are not druggable. So we cannot really use for treatment. We have to find maybe 10, 15, 20, those which are of importance. And another observation is that we see changes. So it's not the same pool of affected genes and proteins in every patient. Always there are some variations. So we hope that with space, we will have additional information to what we have already on Earth. This is about science. And then about another idea of this poster. When we will have cells back, it is a resource for cooperations. We cannot do everything in my lab. No, we focus on proteomics, systems biology, biochemistry, but there are many, many other things which one can do. And therefore, another idea with this presentation is from my side. Maybe someone would be interested to look on biology from point of view of medical support of space missions, development of technologies here on Earth, and maybe some basic science. How living system react in space compared to how this living system are reacting here on Earth. It's a different aspects compared to physiology or medicine, because a lot of things have been done so far as well. And I think time is ticking, so if there are questions, I would be happy to answer.
Time is ticked, and that's all the questions. I apologize for
the
missed pronoun earlier, but thank you very much for bringing this in.
Thank you.
I appreciate it. Going on to our next poster. Next poster is the effect of sowing density on the reproductive cycle of rapid cycling fast plants, Brassica Rapa L. Jolynne Woodmansee, BioTECH Science faculty at the BioTECH Highschool, is the presenter.
3, 2, 1.
Introduction. Growing Beyond Earth is a collaboration between Fairchild Tropical Botanic Garden and NASA in efforts to find suitable food plans for future space flights. We began our project last year at our school, BioTECH High School in Miami, Florida. However, due to COVID-19, we had to finish the remainder of our project virtually. We replicated a ground control to astronaut-like setup where we designed the project and communicated with our teacher, who then carried out our requests.
Objectives. The main objective of GBE is finding plans suitable for future space flight. For our project specifically, it is to provide an alternative seed source for extended space travel.
Methods and materials. Our project focused on using the stress of a high sowing density, or the amount of seeds planted in a specific area, to trigger more rapid reproduction and increase the production of seeds. If successful, this method could be used to provide for future deep space missions where plants would be grown specifically to provide seeds. In order to test this in conditions best replicating the ISS, we developed an experiment using rapid cycling Brassica Rapa. These plants were chosen for this trial due to their 40-day life cycle and the previously successful growth of the Brassicaceae family on the ISS. In this experiment, we used 2 sets of grow units of different sizings and LED lighting. One set of grow units had a higher PAR and larger area to allow sufficient watering through wicking mats. The other set had a lower PAR and much smaller area, causing water stress. Each set included 2 grow units with 6 pots in each, differing only in sowing densities, being a control group with 1 plant in each pot, and an experimental group with 4 plants in each pot. Throughout the experiment, data was collected to track the plant's growth in response to sowing density in each of the stages of development, including germination, vegetative growth, flowering, and seed harvest. In order to promote the development of seeds, we cross-pollinated the plants within each unit with a cotton swab, as shown in Figure 2. At the end of the experiment, we harvested and measured all the pods and seeds, as shown in Figure 5.
Results and conclusion. The final comparison of harvest data per treatment was recorded, which included inflorescences, pods, pod length, and seeds, as shown in Figure 6. It was found that the second treatment with the higher sowing density induced almost double the amount of pods per plant, with 3 times the amount of seeds per plant. It was also found that the grow units with the higher PAR had higher control consumption and masked the effect of planting at higher seed densities. Based on these results, the most optimal method of growing for seed harvest is with higher sowing density under lower light and induced water stress. In the future, if this project were to be replicated, extra dwarf pak choi would be used because it is the candidate species already used on the ISS.
Live. Okay, bring her in. All right, so thank you very much, Jolynne. It's time for us to have some Q&A with you. We do not have any existing questions at this moment. So why don't you go ahead and spend a couple of minutes telling us more about your project, and I will interrupt you if we need to when questions come in. So go ahead and take the stage, it's yours.
Okay. My screen is blank in front of me, so are you okay on your end?
We're working on getting it back up. Some of the files are difficult because of the way they're stored.
Introduction. Growing Beyond Earth is a collaboration.
We want to pause. There we are.
And is there any way to bring in my students to answer the questions?
No, there's no way to do that.
Okay, because they're way better prepared than I am to answer them. Is it something they could type the answers in and I can read their answers?
No, there's no way for us to do that.
Okay.
We have another 5 minutes for you.
All right. Well, this project involved some very perseverant high school students who were very fortunate to have Fairchild Tropical Botanic Garden as part of our school's program. And so we work there with Marion Litzinger, who runs the Growing Beyond Earth program there, which involves over 150 schools in Dade County. Our school specializes because we're on site, so we have access to their labs and their resources. And so this is a conservation magnet high school that is involved in helping through the Growing Beyond Earth program better understand how to grow plants in space, both aboard the ISS and for future space exploration. So this project involved repeated trial and error. The data that's presented in this poster was their third attempt and successful attempt at being able to grow these plants throughout the experiment. Involved they basically I mean, they already explained their methods in their poster, but I guess what I would highlight about this is that the interesting results are that they were able, you can see in figure 4, which shows the two setups. The ones on the top represent an older version of the growing units that are modular setups for the veggie program on the ISS. And one on the left had a single plant per pot. The one on the right had 4 plants per pot. The one below is a newer, smaller modular setup. And so they basically ran identical experiments with respect to the number of seeds planted per pot. And they found that basically under stressed conditions of sowing density, in addition to water stress and in addition to light stress, so reduced light availability, that the plants that were planted under higher densities produced more seeds, twice as many seeds. So that was our most significant result, which is the bottom setup in the figure 4 with the higher sowing densities. So that was a very interesting result. And I guess the group concluded that if they were to move further with this, they would try it with the dwarf pak choi, which is already being grown in the veggie up on the ISS. We had to substitute a very fast growing brassica of the same species, which generally is used for classroom set research out of the University of Wisconsin, but we use those seeds in order to have time to complete the trial experiment.
All right. Welcome, Jolynne. Thank you very much. We appreciate your jumping in and doing things off the cuff. I understand how difficult that can be sometimes, especially in today's COVID environment. So I'm going to go ahead and transfer control over to my buddy Sheng-wey. Jim is the executive director of the American Astronautical Society, and he'll be taking the next few slides.
Thank you.
Great, thanks, Alan. And Jolynne, before you go, there is one question that just popped up in the chat. You can type in your answer there directly to that person, I think, if you're capable of that.
I think we've got a minute, Jim. Let's go ahead and let her do it live if she can.
Okay, good. Yeah, go ahead and answer if you want to, Jolynne.
I would actually rather have one of my students type in an answer. Are they able to type in answers?
I don't think they are, not if they're not panelists, sorry.
Okay.
I'll leave it up there and maybe you can get an answer from them and type it in yourself.
Yes, we'll work on that. Thank you.
Great, thank you. Okay, thanks, Alan. Moving on to our next poster. The poster is called Observations of Ultra-Cold Gases in Microgravity-Enabled Shell Potentials with the Cold Atom Lab aboard the ISS. The poster presenter is Nathan Lundblad from Bates College. Unfortunately, I don't see Nathan in the queue. So Nathan, if you can hear me, just let us know that you're out there. But in the meantime, we'll put up his poster and it does not have any narration. So just take a good read of that and we'll be back with you in a couple minutes. Okay, it looks like Nathan has joined us. So Nathan, if you're ready, please turn on your mic and your camera. All right, there you are, sir. Glad you could make it. Can you hear me?
I can, thank you.
Okay, great. So you now have the next 7 minutes to talk about your poster, talk about the Cold Atom Lab and everything you're doing. And if any questions come up, I will interrupt you and ask them. So take it away.
Great. So I'll talk pretty informally about this. This is not a conference that I'm actually super familiar with because by profession or by field, I'm a low temperature atomic physicist. But I have the good fortune to be a user of the NASA Cold Atom Lab, which is currently in flight and in operation aboard the International Space Station. So the Cold Atom Lab, which is built by JPL and NASA, makes ultra-cold samples in microgravity that allow us to explore physical phenomena that we can't actually observe or even try to observe on Earth, basically by going to a free fall or a perpetual free fall environment. Now, a common misconception with a lot of this when I talked about this experiment is that we're requiring the low temperature of space or the vacuum of space to see these effects, but the hardware, the Cold Atom Lab itself, is a rack mount hardware in the shirt sleeve environment of the space station itself. So we really are just exploiting the free fall atmosphere of the space station to do this stuff. The particular experiments I'm interested in are using this environment to start off with what's called a Bose-Einstein condensate. It's a state of matter discovered in 1995 and now seen in a few hundred labs worldwide, where below a certain critical temperature, all the atoms that are trapped in this essentially a magnetic bottle within the apparatus all share the same quantum wave function. They behave as a single superatom in some ways, and they exhibit wave-like behavior, which is not normally thought of for what we think of particles in a gas. But importantly, once you have a gas this cold, it's an exquisite sensor of electric and magnetic fields. It is a remarkable state of matter that essentially, by being that cold, allows you to probe finer and finer degrees of freedom and possibly look for new physical phenomena that haven't been seen before terrestrially. So in particular, the experiments that we're building with my group here are to convert a typical Bose-Einstein condensate, which forms as kind of essentially a ellipsoidal blob, like a rugby ball or football shaped, and deform it into a bubble or shell. So the important thing about atoms living on a bubble or a shell surface is the physics is a little bit different. The topology of the surface of a ball or an ellipsoid is fundamentally different than just atoms moving kind of at the bottom of a well. So our goal here is to see shell-trapped Bose-Einstein condensates or bubble-trapped or bubble-like Bose-Einstein condensates and explore some of their properties. So far what we've been able to do in the first year of running with the ISS and the Cold Atom Lab is to inflate a typically maybe 10 micron size cold atom sample, the initial maybe 100 nanokelvin thermal cloud, and inflate it to the lower left in that poster there, maybe about a couple hundred microns or maybe even 500 microns large. And we wouldn't be able to do this on Earth for the following reason. If we imagine a soap bubble with if the soap film got dense enough that it was actually affected more by gravity, you actually wouldn't be able to form soap bubbles on Earth. They would just sag to the bottom of the bubble and it would pop. And the same thing happens with Bose-Einstein condensates. When we try to make these bubble-shaped condensates on Earth, they pop. You form little contact lens or dish-shaped bubbles in the trap, and you don't form a bubble. But in free fall, and you can see a hint of this at lower left, you can begin to see what look like bubble structures. Now, this is a shadow image. So it's integrated along one direction. But if we had taken these pictures on Earth or in a terrestrial lab, you really would just see a very, very thin line or small dot at the bottom of those pictures, or the bottom of those structures there. Being in free fall allows the atoms to curve up around the sides of the bubble. So we've done some thermometry of these bubbles, looking at the temperature of the bubbles as they expand, looking at some residual micromotion, and are the bubbles actually sloshing around in the trap as we go. But really the next steps we're looking forward in the next year of the operation of the Cold Atom Lab above ISS is to begin to look for signs of sort of the different physics of what's different when atoms live on the surface of a bubble, namely new degrees of freedom for collective excitations, how the whole thing sloshes in a different way than it would on Earth, but also look for signs of vortex behavior. So ultra-cold systems like this can develop quantum vortices, and they behave very differently in a curved environment like a shell or a bubble than they would in a typical trap. And that's what we're after with this. And I think I'm a little ahead of my time here, but I'll stop talking there and see if there are any questions or not. Thank you.
All right, thanks, Nathan. Yeah, one just came in just this instant. So if you could be real brief, what characteristics of ultra-cold atoms or gases do we study?
Yeah, that's a great question. I think typically some of what we study right away is at what transition temperatures do they begin to develop macroscopic quantum behavior? So when do they actually become dominantly wave-like instead of particle-like? But also once they're that temperature, and it's akin to like a superconducting state, how can we use those ultra-cold samples like the Bose-Einstein condensates to make precision sensing measurements of electric or magnetic fields? And so we study their sensitivity to magnetic fields pretty extensively, as well as electric fields.
All right, great stuff. Hey, Nathan, thank you so much for being here. Thanks for presenting. It was great stuff.
All right.
Appreciate it. All right.
Hey, everyone. My name is Brendon Ortolano.
Oh. Okay, time to get started with our next one. This is Crystal Growth of Metal Organic Frameworks in Microgravity, a case study of reaction chambers for Cu9, Cl2, CPA6. Brendon Ortolano from the University of West Florida. Go ahead with the poster.
A mechanical engineering senior from the University of West Florida. Today, my two teammates and I will be discussing the Tesseract, a nanolab designed to grow the metal organic framework Cu9Cl2 CPA6 aboard the ISS in microgravity. At the top left, you can see our abstract, in which we describe in detail the material we're studying, as well as some important characteristics of both the synthesis vessel and the material itself. Cu9Cl2 CPA6 has a molecular structure known as the triangulated Kagome lattice, or TKL. Pictured on the left, you can see what happens when ions try to assemble in a triangular shape. One pair of ions will be unable to order, resulting in geometry that is frustrated. This applies to more complex structures too, such as honeycomb geometry, the Kagome lattice, and, in our case, the TKL. In Figure 4, you can see the molecular lamination of our material, which causes further interactions between ions. This frustration provokes the material to exhibit important characteristics, particularly what is known as the magnetocaloric effect at cryogenic temperatures. This is due to the material's inability to magnetically order. When the material is in an unordered state, a magnetic field may be applied to order the material and reduce entropy, allowing cooling of the crystals by repeatedly applying and removing a magnetic field. Because our material remains unordered and retains finite entropy down to 50 millikelvin, it has the potential to work as a cryogenic solid refrigerant, giving it possible space applications.
Hello, I'm Joseph Lupton, a senior mechanical engineering student at the University of West Florida. I'm here to talk a little bit about the reaction vessel requirements. So to complete a synthesis aboard the International Space Station, the reaction steps had to change from something that a human can do to something that can be done autonomously. The main changes from a lab-based synthesis to one done in a microgravity environment are filtering, powder addition, and stirring. Without gravity, all of these things become very difficult to perform due to adhesive and cohesive forces. Primary restrictions that we faced, including volume, power consumption, and power loss, were addressed through mechanical and software design. To prevent power saturation, a Python script was developed to run each subsystem in a distinct sequence. The program also has the ability to keep track of what steps the synthesis is on and how long it's been there, so the synthesis can pick up where it left off in the event of the power outage. The original concept for the Tesseract revolved around the use of different subsystems to perform the synthesis of different MOFs. The subsystems that have been created for this case study have the ability to filter, pump, stir, and isolate caustic materials. When these subsystems are paired with Python script, which has the ability to run all of these processes without pulling too much power, multiple types of MOFs can be created.
Hello, my name is Jacob Moses. I am a senior mechanical engineering student at the University of West Florida. I will be talking about our various subsystems. Leaving the atmosphere introduces many variables that are not typically encountered on Earth. That being said, we opted for as many reaction vessels as volume permits, resulting in chambers, filters, and pumps. This allows us to synthesize on a scale that is known to produce a controlled reaction. Smaller reactions have been easier to handle and provide more consistent results. The pump mentioned next circulates a copper 2 chloride solution through the 4 chambers and filters. Each syringe is actuated to inject sodium hydroxide into the chambers, resulting in a precipitation of copper 1 chloride, which is then slowly filtered off. Once filtration is complete, the solution will rest in the chambers until re-entry. One of the most crucial systems for the Tesseract is the ability of fluid control. These pumps allow fluid to be sealed off during transport and also allows forward and reverse fluid flow. As for the peristaltic pump, this form of pump was most ideal due to its sealed nature when inactive. It works similar to human organs which use a pinch and push motion. This allows fluid to stay contained during transportation. These pumps are typically used in medicine because the fluid pump does not come into contact with the pump itself, allowing for the flow of caustic solutions. Our team opted to design our own peristaltic pump so the flow rate could be maximized under a very specific dimension. This pump is perfect for nanorack utilities due to its ability to stack for a variable number of synthesis trials. It can be paired with sensors to have precise data acquisition on the flow system. Each of these subsystems have unique design criteria to withhold and tasks to perform in conditions not seen on Earth. Specific data on the subsystems can be seen at the top right of the poster. Data including weight, flow rate, power consumption, and controllers used. In conclusion, we as a team strive to make an all-in-one package that has potential to be used in many other applications aboard the space station. Further testing shall be conducted to fine-tune our product to ensure efficiency in our design. Thank you for your interest in our work, and if you have any questions, feel free to send an email to the address on the bottom of the poster.
All right, very cool. Brendon, please bring yourself up on screen. There you are. How are you, Brendon? Oh, I can't hear you, unfortunately. Does anybody else hear Brendon?
Can you hear me now?
There you go. There you are.
I was trying to use a separate mic, but I guess it's not working right now.
Technical fun. All right. So, Brendon, there aren't any questions for you right now posted online. Anybody out there in the Zoom room, please don't hesitate to ask any questions. But Brendon, you have the floor to talk to your poster for the next 5 minutes.
Okay, I do want to start off by saying I'm sorry some of the text is a little small. We started off in Google Sheets. We wanted to do the full 8-foot poster that you guys had previously required. And then when we brought it over to PowerPoint, we had to shrink everything down. So everything is a little small. So yeah, this is a material that Dr. Terhar at UWF has been studying since the early 90s. I want to say 92 when he was at Bell Labs. And then when he came back to the University of West Florida, he brought it with him, and chemists at our university have been doing some tests on it for a while. That's where we get all of the data seen on the left. And then where the engineers come in is we wanted to explore how this would actually function and grow in space. And we wanted to get some samples that were uniform and stable, so that way we could do some additional materials testing, figure out all of the variables just about the material before we send anything up into space. So we currently have completed our first prototype, which is just to serve as a proof of concept. We modeled it in Onshape CAD software. Those models are what you see in the center of the page. And these include different subsystems that have that you can see on the right over here. We try to make the poster as comprehensive as possible. So again, I'm sorry if you can't read much. If you're interested, please take a look at this again later and email me if you have any questions. Do we have any Q&A?
No questions for you right now, Brendon. You're free to talk some more, or we can let you go and we'll take a little break before our next poster. Up to you.
Okay, I think that's about all I've got. Like I said, I did try to make the poster as comprehensive as possible. My email is at the bottom right if you guys have any further questions.
Great, and thanks so much, Brendon. Everybody out there can go on the website and download these posters themselves, and so that's an opportunity to expand and look carefully at stuff and get your email and everything like that if they do have any questions. So thank you so much, Brendon. Thanks for being here. Thanks for being a part of it. All right, it's 12:48. We're just going to take a 2-minute break, and we'll be right back with our next poster. Okay, that was a nice short little break there. Let's get started with our next poster. This one is direct detection of Chameleon and Symmetron dark energy in microgravity. Sounds really cool. And this is brought to us by Sheng-wey Chiow from the Jet Propulsion Laboratory. Let's run the poster.
90% of the universe is composed of dark energy, which drives the accelerated expansion. But no one knows what dark energy is. Traditionally, dark energy is studied through astronomical observations. Bigger telescopes allow us to look further back in time, so we can learn the evolution of dark energy on the cosmological timescale. However, better observations will not likely lead to distinction between dark energy models. We propose to test dark energy scalar field models directly in lab. In fact, the most stringent constraints so far are placed this way. How do we do it? First, we start from dark energy models of interest, specifically the Chameleon model and the Symmetron model. By solving the equation of motion of the scalar field, we can predict the dark energy force on an object, which depends on the parameters of the model. The force, or the acceleration here, is proportional to the gradient of the scalar field, but suppressed by a screening factor. This screening factor is very small, and that explains why we don't see the effect of dark energy in our daily lives. However, for a small particle like an atom, the screening factor is 1. That is, single atoms are much more sensitive to dark energy fields than bulk objects. And it happens that atoms can be used as a very sensitive force sensor. The technique is called atom interferometry. In an atom interferometer, the metal wave of a single atom is split and recombined to interfere. The interference phase is determined by the potential that the atom experienced during the operation. Atom interferometer has several unique properties. For example, it has precision and accuracy. for direct dark energy detection. However, the most important aspect is that it is a measurement using single atoms. Previously, atom interferometers have been used to constrain Chameleon and Symmetron parameters, as shown in the shaded regions. Our project will extend the bounds of the parameters by at least a factor of 10. The major limitation of detecting dark energy using atom interferometer is the gravity. The expected dark energy signal is weaker than gravity, even for atoms. We take the advantage that we know the equation of motion of the scalar field so that we can design the scalar field to minimize the gravity effect. Intuitively, the dark energy field is smaller near an object and larger in vacuum. If we have a periodic cylindrical structure, the scalar field inside is spatially modulated. This is shown on the figure with a false color field intensity profile. You may guess that the gravity from the structure is also spatially modulated. That is correct. By adding the red 3 rings, we can double the modulation frequency of gravity, but leave the scalar field largely unaffected. Now, if we perform an atom interferometer that can pick up periodic potential signal along the axis of a cylindrical structure, we can detect only dark energy forces but not gravity forces. This experiment has to be conducted in microgravity, so that atoms can move slowly and steadily through the structure instead of falling through. The Einstein elevator at University of Hanover in Germany is a recently completed facility. It can provide 4 seconds of microgravity time and 100 runs a day. We are collaborating with Professor Russell at University of Hanover for a project called Direct Detection of Dark Energy on Einstein Elevator. We believe that we can improve the constraints of Chameleon and Symmetron by a factor of 10 in 3 years. Of course, the ISS is a much better microgravity platform for this kind of experiments. You will learn more about cold atom experiments in other presentations in this conference. The Cold Atom Lab can be considered as a pathfinder for a future dark energy experiment on board the ISS. Thank you.
Excellent, thank you. Intriguing stuff. I have one quick question for you, Sheng-wey. So bring yourself up on screen if you would.
Hello.
There you are. How are you, sir?
Pretty good.
Good. How deep or tall is the Einstein elevator at the University of Hanover?
I think it's a few meters deep and tall, maybe 20 meters tall.
Okay. And it still gets 4 seconds. It's pretty good.
Right. They launch from the basement and then they let it fall back. So it's 4 seconds, free fall time.
Gotcha. Very cool. So we don't have any questions as of yet from the audience. Please, audience, if you do have any questions, please put them in the tool. Oh, and actually one just came in. And this is a great question, actually. What is chameleon gravity?
Okay, so chameleon is a name for this particular scalar field model. It's named chameleon because chameleon changes color based on the surrounding. And that is the property of this scalar field, because we don't really see dark energy in our daily life. So we think, or people say that a valid theory must hide the dark energy itself around the surroundings. That's why this theory is called chameleon field. Yeah, the specific expression is at the lower left of the poster, but yeah, the name is just a name saying that it's sort of blending with the environment, that we don't see it. And we try very hard to see that using atoms.
Excellent. Okay. So there are no other current questions. So feel free to talk a little bit more about your poster. And if any other questions do come in, I'll just jump in, interrupt you, and present them to you.
Sure. So I can talk a little bit about these astronomical observations. I heard that there are $1 billion missions is being constructed to see better further into the universe. But there is still a limitation on how far there's an ultimate telescope that people can build, because there are just this many stars. This far we can look back. Beyond that point, there's no more stars. So there's a theoretical limitation on how good we can do the observations for dark energy study. So, and this kind of alternative approach to study dark energy is kind of intriguing and interesting. So I think this is kind of it's actually a new field, and it's a lot of people are actually looking into this kind of alternative approach of dark energy study. And another sort of story of the Einstein elevator is that the Einstein elevator is built on the campus of University of Hanover. Because of the city code, they say that, well, you cannot build anything taller than, you don't want to create another scene. So the building cannot be seen from outside. That's why the height of the building is such that that's why it's not the 10-second tower. It's only 4 seconds, because it had to be short enough that people outside the campus cannot see it. But anyway, it's just some little stories. And another thing is that I think people probably heard of the quantum initiative, that the US is trying to push the quantum technology in many fronts. And atom diffraction is actually one of the sort of quantum sensing aspect. And for the quantum sensing to really work, you really want to put the sensor in microgravity so that we have long interrogation time and all the good stuff. So I think this is a good first step towards this making quantum sensor really useful in space and microgravity environment.
Excellent, Sheng-wey. Thank you so much. And yeah, that's interesting that the tower can only be that height just because of its location. It's like a lot of our buildings here in Washington, D.C. They can't be above a certain height. So definitely
Yes.
appreciate that. Well, very good. Well, thank you very much for your time. We appreciate your being here. And
thank you.
And I just want to remind everybody that, oh, actually, we just got one more quick question for you. If you can give a very brief answer.
Sure.
Does the magnitude of chameleon gravity change with respect to time?
We don't study the time dependence. That's why this equation on the lower left-hand side is static. It's time independent. We think that the time evolution is on the say cosmological time scale. But if the source mass is actually changing in time, then the field will change in time. But it's sort of instantaneous instead of the parameter changes in time.
Okay, very good. Again, appreciate your time today, and very informative stuff. We appreciate it. Okay, so just for everybody's reference, you can go to issconference.org/agenda, and you click through there, and you'll see the posters, and you can view any of these posters at your own on your own schedule, and look into the details. I know it's a lot of it's tough to see here online, so we encourage you to go look at them that way, and you can get every bit of detail that you want. Our next poster was actually rescheduled to 4:10 Eastern today, 16:10 Eastern. So we're going to take a break, and we'll be back at 1:10 Eastern. Thanks very much.
All right, so our next presentation is observing the random dispersion of compartmentalized droplets in an oil-water emulsion in microgravity. This poster is by Jennifer Song and lots of other people at Valley Christian High School. Before we play that, I would like to really remind everybody to please jump in and enter questions as quickly as you can so we can provide the questions to the presenter. We really cannot exceed the 10-minute time that we have for each poster. So if we can play Jennifer's presentation, we'll move on.
My name is William Tang from the Valley Christian ISS program. Our experiment is observing the random dispersion of compartmentalized droplets in an oil-water emulsion in microgravity, and flew on the ISS in March of 2020 under the mentorship of Marcel Mark. We give special thanks to Dr. Thomas Russell and Dr. Paul Kim. First of all, background research. Interaction forces differ between two different liquids, resulting in change of interfacial potential energy as molecules are moved towards a border. Work must be done against the unbalanced interaction forces to bring the molecule to the interface. To reduce potential, liquid structures tend to minimize surface area by forming spherical domains, coalescing, or observing particles to the interface. Polyacrylic acid, PAA, which exhibits low binding energies and a slight negative charge, is dissolved in the silicone oil. The inherent negative charge of the water-oil interface hinders the polymer from binding to the interface. Furthermore, the low binding energy of the nanoparticles prevents the formation of an irreversible bond. To anchor the charged PAA to the interface, POSS nanoparticles with a positive charge is mixed into the oil, forming a nanoparticle ligand assemblies at the interface by electrostatic attraction. The absorption of the nanoparticle ligands at the interface, in addition to the droplet's tendency of lowering interfacial surface area, irreversibly jams the particles in place, forming a flexible, permeable structure with special chemical properties. The structure can then be modified by injecting or reducing the volume of the droplet, shining a light of special wavelengths to weaken the interfacial membrane, or adjusting the pH level of the liquids to further change the characteristics of the structure. Our experiment, a solution of PAA in water is initially injected into a sealed chamber, with capillary tubing connecting to a bidirectional pump protrudes into the chamber, functioning as an injection needle. A gridded sheet of paper is attached onto the polycarbonate sheet in the back of the chamber. An LED is placed behind the chamber, and translucent grids are used as a reference to show how light is refracted through the droplet. By turning on the pump, a solution of nanoparticles and silicone oil is injected into the PAA solution, forming a spherical droplet 2 millimeters in diameter at the tip of the needle. The oil pump then reverses, allowing the oil droplet to shrink for a small interval of time, and the LED turns on again. A picture is taken after each reduction in volume, giving time for the droplet to set. The entire experiment is then looped for the duration of the timing test frame, with 7 1-second interval injections followed by 7 1-second interval retractions. Each injection interval increases by 30 milliseconds per loop, while the retraction intervals remain constant. Our hypothesis is that if we print droplets of different liquids in a second host liquid that are stabilized by nanoparticle surfactants in a microgravity environment, then a random arrangement of droplets can be achieved due to the lack of buoyant forces, with on-demand control over coalescence. To analyze our experiment, as observed, our experiment functioned. For the first 14 cycles, the injected droplet can be seen oscillating on the needle and slowly increasing in size. However, after several cycles in which we were able to observe a slight wrinkling of the membrane, the droplet volume continued increasing, such as it adhered to a panel of the chamber by surface tension. An intriguing phenomenon was observed. Interestingly, when the needle was attached from the oil droplet, it retracted liquid from the surrounding chamber during a retraction interval and subsequently injected streams of small water droplets into the larger droplet of oil. These smaller droplets dispersed into the surrounding oil, eventually stabilizing in individual locations. Coalescence was not observed, and the droplets flowed independently of each other in the liquid of the large droplet. On Earth, due to density differences, the buoyant forces would have caused the water droplets to accumulate at the top of the larger host droplet. Individual droplets would coalesce before a membrane could be formed of the POSS nanoparticles and PAA at the interface. Thus, it is extremely difficult to form the compartmentalized all-liquid systems on the ground, or observe the interaction between interior liquid fluid interfaces and structures. On the ground, we were able to obtain valuable data through testing as well. Due to gravity, silicone oil droplet, denser than the aqueous PAA solution, sag downwards, falling off the injection needle regardless of chamber orientation. Overall, our experiment was operational and yielded some very interesting results that may be of use in the development of all liquid encapsulation systems. We were able to evaluate our hypothesis with the data collected and use ImageJ, an open source image processing package, to identify trends, take precise measurement, compile animations, and calculate the area of a droplet, in order to convert the large volume of data recorded, 1,271 photos, into a visual form.
All right, That's a lot of material in 5 minutes. Sounded like the end of a pharmaceutical commercial, but it was understandable, and the diction was very good. So thank you very much for helping us get all that information. We have Jennifer Song, and Jennifer, at this moment in time, we don't have any questions. So what we'll do is we'll let you talk off the cuff, ad-lib a little bit about your poster and your team, and you can do that for the next 5 minutes. And if we have a question come in, or when we have a question come in, we'll interrupt you and bring it to you.
Sure. So we are actually a team of students at Valley Christian High School, where we have a program called the ISS Research Lab. And here we have the opportunity to design and construct our own experiment to fly on board the ISS with the help of some very knowledgeable mentors. And then for this specific experiment that we did, we reached out to Professor Thomas Russell and Dr. Paul Kim from Lawrence Berkeley National Laboratories. And we were honored to receive a lot of their support and guidance as well. So for this experiment, since there are no questions so far, essentially we are observing like the assembly of nanoparticle ligands in between like a liquid-liquid interface. So here we have water and silicon oil, and in the water is mixed PAA, polyacrylic acid, which kind of acts as a ligand that grafts onto the nanoparticles, POS nanoparticles, which are dispersed in the silicon oil. So when that happens, the ligands and the nanoparticles meet at the interface, and that kind of creates a membrane that separates the oil from the water. And this can be used in a variety of applications, such as being able to have compartmentalized reactions in these different encased liquids. So basically all liquid capsulations that you can activate on demand. For example, if you introduce like higher pH, if you change the pH of the surrounding liquid, if you shine a light of a specific wavelength on this bubble, for example, then two different bubbles with two different reactants can combine and interact because the like the light or the pH level can change the properties of this membrane, and that can be used in biological experiments, for example, drug encapsulation, all liquid batteries, and a variety of applications.
All right, we still have no questions. So a question for you from me. What is this a multi-year project, or is it a single-year project?
This is a single-year project. So we started near the beginning of last school year, and this flew on the ISS in, I think, March of 2020. And this year we are continuing this experiment to really focus on the kind of compartmentalized reactions. So we're observing the diffusion of two different types of reactants across this barrier, across the membrane, and seeing how these properties of diffusion are affected by microgravity.
All right, and how has COVID affected how you're able to do your work?
Yeah, so right now, all of our work is done online through Microsoft Teams. So we do have meetings twice every week, and that's how we are currently planning and designing our experiment with the students and mentors. And then I think we're planning to have like kind of drop-off boxes at our school where we're able to get all the materials necessary after sanitizing it, and then work at home and then bring it back to school.
Oh yes, sanitize everything. I understand that. Well, Jennifer, thank you and your team. We appreciate you bringing this to us, and we hope that you continue with your studies very well. Even if you have to do it at home with your parents watching.
Thank you.
Okay, thank you. So moving to our next effort. There we go. Moving to our next time, we have neural stem cells proliferate more while in space and are larger upon return to Earth. Implications for astronauts' health. Well, we have the presentation. It is a silent presentation, and the presenter has not come into the green room yet, or the waiting room for the sessions. What we're going to do is we're going to bring up the presentation for 3 minutes in silence. And at the end of 3 minutes, if the presenter is there, we'll let her jump in. And if not, then we will go ahead and take it down and take a break until 1:20 Eastern time. So if we could, can we play the presentation? All right, so a few folks read the presentation, and we even had a question come in. I'm sorry that the presenter is not here. So what we'll do is we'll take a little break and come back to you at 1:30 with the next presentation. Thank you.
All right, and welcome back. We're ready to go with our next poster here. This one, I get the pleasure of saying it's Cosmic Ray Energetics and Mass for the International Space Station. The acronym is ISS-CREAM. Interesting. So, and this is presented by Yoon Suk Seo from the University of Maryland. Let's go with the slide.
Hi, welcome to the ice cream poster. Yes, you heard it right. The project name is ice cream. Yeah, it's written as ISS cream and pronounced as ice cream. That's cream for the International Space Station. Here, CREAM stands for Cosmic Ray Energetics and Mass. The CREAM project is studied as a balloon experiment. As you may have guessed from this photo at the bottom of the poster. This balloon-borne CREAM was flown successfully 7 times over Antarctica, accumulating total about 191 days of flight time, which is the longest exposure for a single balloon project. Building on this success of balloon flights, the payload was transformed for accommodation on the space station, specifically NASA's share of Japanese experiment module exposed facility, GEM-EF number 2. So this is an external payload. This version of CREAM called ice cream was launched aboard the SpaceX Falcon 9 rocket as part of a CRS-12 mission to the station on August 14th of 2017. Since its successful installation, the ice cream experiment collected data until it was shut down on February 12th of 2019. The ice cream payload is about the size of a refrigerator, but it's heavier with a mass of 1,258 kilograms, as shown in the middle of this poster. The ice cream instrument is configured with complementary particle detectors to determine the charge and energy of high-energy cosmic rays from protons to iron nuclei. And the goal of this experiment is to extend the direct measurements of cosmic rays to the highest energy practical, so directly measuring the composition of cosmic rays. Now here the flight data show the instrument performed rather well. Individual elements are clearly identified with excellent charge resolution, as illustrated at the top right of this poster. The calorimeter provided instant particle energy as well as trajectory measurements. The preliminary analysis shows the power law nature of the cosmic ray spectra clearly, and individual elemental spectra are being analyzed to address detailed spectral features. Current analysis indicates that the data extend well above 10 to the 14 electron volt. This is a humongous energy. At these high energies, cosmic rays can generate gigantic air showers. That's the product of the interactions of primary cosmic rays with the atmospheric nuclei. And the showers, they can reach all the way to the ground. And so they have been mainly measured or observed indirectly from the ground. So it's very exciting to see these ultra high energy cosmic rays in space, to measure them directly before they break up in the atmosphere. Our findings at these high energies will provide keys to understanding the cosmic ray origin, acceleration, and propagation. Stay tuned. Thank you.
All right. Thank you so much. Very interesting stuff. That was a really long duration for that balloon, total duration for that balloon. That's amazing. It's like more than half a year. So, Yun, sorry, I'm not sure how to do your first name, but please bring yourself up on camera. And let's see. There you are. Excellent. Good. Well, good to have you here. Thank you very much for the presentation. And we don't currently have any questions for you from our viewers. So please feel free to take a few minutes. You have about 4 minutes to talk more about your project if you want. Otherwise, just turn it back over to me and we'll go from there.
Sure. Well, it's not my regular conference, but obviously, but I wanted to give a status report because I was invited to this conference, the ISSR&D conference, the first one year, let's see, was it what 2012 or so, to talk about plan. So at that time, we had a session on space science for the ISS, and I talked about my plan with this project at that time. And now we've flown, and so I thought I should report to this community. Oh yeah, I should actually mention that I've organized a couple of sessions for this kind of predecessor conference. The name was a bit different back then, about 20 years ago now. What was that? The STAFE, as I remember, it was a STAFE, Space Technology, Space Technology and Applications, maybe the International Forum or something. So so at that time, we discussed about the space station utilization. So I organized a few sessions at that time and planned for missions, and it took a long time to realize the mission. And obviously, a lot of people worked very hard for a long time to get a mission done. And although this conference is not my typical physics conference where I would discuss much more details about the result and the cosmic ray physics, but rather here I wanted to give a status to let those who supported the mission along the way for a long time. You know, there are so many people in so many aspects to support. And this conference, I know some of people, some engineering folks attended this, so I thought I would just give a report. So, and I would like, so, you know, take this opportunity to thank those who helped us and supported us and worked so hard to make this mission realized. So, and also let me thank all the organizers who worked hard to organize this conference, particularly first time now virtual online conference, right? So yeah, it must be very challenging. And I'd like to give you an encouragement for that matter to make this happen. You must have worked hard to do that. And yeah, so thank you, everyone.
Great. Well, thank you. And we actually do have one question that came in, so that's great. So could plants live more than 6 months exposed to these cosmic rays? And the person says, think of lagoons and Crassulacea plants.
Well, I would not know about plant. Yeah, I can only say that cosmic ray deionized particles that, you know, affect so many different things, obviously. You know, it affects the structure of the galaxies, evolution of stars, everything, all these living things on this earth, their evolutions all get affected by it. But, you know, depending on what plant where exactly, and and so on. On this Earth, obviously, with the radiation that we have, which is protected by the atmosphere, so everything is flourishing. And outside of this atmosphere, where you don't have such protection, you are in trouble. And particular plant that must be asked to a plant scientist, yeah, whether that can survive or not. And yeah, I mean, for plant to survive, not only radiation or issue, but also you have to have fundamental basic things, right? Water and the solar, sunlight, etc. So it'll be much more biological question than this cosmic ray question, I would think, for that survival of a plant.
Understood. Well, great. Thank you so much for your time today. And thank you for coming back and updating us on this. It was great to hear that you were at the original ISSR&D conference. That's amazing. That's great. And I think we should do more of that, bring people back to after their projects and experiments have flown. So I think that's really neat. So,
all right.
Excellent. Thank you so much.
Sure. Okay. Good luck. Bye.
Thank you. Okay, everybody, we're going to take a break now because our next poster was withdrawn. So we will be back in about 9 minutes at 1:50 Eastern for our next poster. Thanks, everybody. Okay, and we're back. Let's do our next poster in our 1:50 p.m. time slot. This one is called 3 Years of Observations from the International Space Station by the Stratospheric Aerosol and Gas Experiment, Part 3. Our presenter for this one is David Flitner from NASA Langley Research Center. Let's roll the poster.
Welcome to 3 Years of Observations from an International Space Station by the Stratospheric Aerosol and Gas Experiment 3, SAGE 3 ISS. From the vantage point of the ISS, SAGE uses the solar occultation technique to monitor the vertical distribution of aerosol, ozone, water vapor, and other trace gases in Earth's stratosphere and troposphere to understand ozone recovery and climate change processes in the upper atmosphere. The why, how, when, and where of SAGE 3 ISS are covered in the left and middle columns of this poster. Moving directly to the right-hand column in the upper blue box are the results related to notable stratospheric aerosol events as recorded by SAGE 3. Since commencing science observations in June of 2017, SAGE has recorded numerous perturbations of the stratospheric aerosol layer. For example, in August of 2017, extreme wildfires in Western Canada caused tremendous heating that produced smoke-laden water clouds, also called pyrocumulus, to rise to the lower stratosphere. Smoke from the pyrocumulus enhanced the stratospheric aerosol by as much as 8 times, and is shown in the figure on the upper right of the blue box. In the middle part of the blue box is a time altitude representation of SAGE aerosol measurements for latitude band of 40 degrees north. The impact of the Canadian wildfires is seen in the first part of the record. In the latter half of the time period, you can see the Ryukoki July 2019 volcanic eruption in eastern Russia that injected the largest amount of sulfur dioxide into the stratosphere has been seen in the past decade. In the stratosphere, the sulfur dioxide was converted to stratospheric aerosol droplets, which are the main component of the stratospheric aerosol layer. The devastating Australian bushfires of 2019 and 2020 produced pyrocumulus effects that dwarfed those seen in the Canadian wildfires. Here we see in the lower part of the blue box an aerosol evolution time series for 40 degrees south since June of 2017. Smoke from the Australian bushfires is clearly evident beginning in January 2020. This smoke lingers still in the stratosphere 6 months after the initial injection and demonstrates the long lifetime of aerosols in the stratosphere. Moving to the lower part of the right-hand column, SAGE has recorded variations in stratospheric trace gases, ozone, and water vapor over the past 3 years. The seasonal cycle is the dominant natural pattern of the amount and distribution of ozone and water vapor in the stratosphere. However, when this yearly pattern is removed from the data, other factors influencing variability can become evident. One such influencing force is the quasi-biennial oscillation, which manifests as a downward propagating anomaly in stratospheric wind and temperature. The impact is most easily seen in observations of the equatorial region and shown in the 4 plots in the lower part of the right-hand column. In the upper right-hand of these 4 is the temperature anomalies as depicted in the NASA mirror product, showing the variation, the downward propagating anomalies in temperature. Across from that, in the upper left, are ozone anomalies, which bear a strong resemblance to the temperature anomalies since ozone is greatly influenced by temperature variations. Similar but not exactly the same as ozone is the QBO influence on water vapor. The lower left and right-hand figures show the altitude time series of water vapor mixing ratio and the anomalies respectively. The impact of temperature anomalies on water vapor first influences the entry of water vapor from the moist troposphere to the dry stratosphere and is captured in the upward propagating wet and dry mixing ratio anomalies from 15 to 27 kilometers. Above 30 kilometers, the variations of equatorial water vapor are dominated directly by the temperature changes, and the patterns more closely resemble the temperature anomalies. In summary, SAGE-3 ISS is a collaborative Earth science climate continuity mission to study the recovery of the global ozone layer and the evolving climate. Since June of 2017, SAGE-3 ISS has documented the many natural variations of important stratospheric trace gases and the perturbations caused by major volcanic eruptions and record-setting wildfires. Thank you very much for your attention.
All right, very good. Thank you very much. Thank you, David. So David, bring yourself up on screen. And while he's doing that, I will tell everybody out there in the webinar to please submit your questions, any questions you might have on this, this interesting project. In the meantime, David, please tell us more. Just talk to your poster and tell us more about the project.
Okay.
So talk about SAGE-3. Thanks.
Yeah, SAGE-3. I've told you about the science results. How we make the measurements initially is basically looking at the sun as it rises and sets with respect to the space station. So with that, we've got a nice bright source and we track it till observing the light as it passes through the atmosphere of Earth. And from that, measuring at various wavelengths, we can deduce the different composition of the atmosphere at different levels. It allows us to do this in a higher vertical resolution aspect and with very low signal to noise because of the bright source. The fact that the ISS has this inclined orbit is really ideal for the solar occultation method because it allows us to sample all through the mid-latitudes through the tropics, north and south, on roughly a monthly time scale, and that's really syncs with the processes that go on in the stratosphere. So it's a good
David,
marriage.
before you go on, we do have a couple of questions that have come in.
Okay.
So the first one is SAGE-3 still going? And if so, has it produced any research with the current Western U.S. fires?
Yes, excellent question. Yes, it is ongoing. Thankfully, we're on the manifest for as long as the ISS is willing to host us. And not presented here, but we just have some recent indications of the California wildfires. We're watching that as that evolves. But yeah, there has been perturbations and signals up in the stratosphere as well. So it's been interesting. Typically, what we see change in the stratosphere is due to volcanic eruptions. As of late, there have been these tremendous wildfires that produce these smoke-laden clouds that reach the stratosphere. So that's been neat to see that and track and see how that evolves.
Yeah, very good. And so I'm sorry if you said this, but does it have an end date right now?
No, it's pretty much open. As far as NASA is concerned, we had a 3-year prime mission. We just had that review actually just last week to close that out, and we had submitted an extended mission proposals to the science director at NASA, and that's under review. We're expecting guidance within the next month or so, but indications are we'll go on, at least have funding for another 3 years for certain.
Another 3 years, excellent. So, so then a follow-up question to that: will there be a SAGE-4? And if so, I'll add on, and you have 1 minute, unfortunately, to answer this one. What would you do differently for a SAGE-4?
So for SAGE-4, actually, we have a project that we're pursuing to make the same science measurements, but at a different form factor. We started, a colleague, Dr. Rob D'Amadeo, looked at things, says, what does he need for measurements? What assumptions is he using in the data analysis, and maybe what different aspects of how we make the measurement could address those assumptions. So actually we're looking at what would be more like a CubeSat type footprint that could maybe be launched from the ISS, but then would be a free flyer. Sometimes people call that sensor craft in a sense, the marriage of the instrument and the craft itself. So yes, we are looking to prolong the measurements, trying to do it in a maybe more economical footprint, but maintain this.
And that's just a real quick answer to this one, if you could, because it's exactly what you were just talking about. are there pros and cons to being attached to ISS rather than being a free flyer?
Yes, yes. A pro is that, you know, for example, the power system goes down. The astronauts can address that. That happened earlier this year. A con happens to be we have to live to what the ISS schedule is with visiting vehicles and this and that. Or we're trying to come to grips with maybe what the vibration environment is. It's not impacted tremendously our measurements, but it is maybe degrades our science quality a little bit. So some of it, we don't control our full destiny by being a hosted payload.
Right.
I'll say that.
Very, very good. Hey, thank you very, very much. I'm all done. I'm going to turn it over to Alan for our next poster. Thanks a lot, David.
Okay. Thank you very much.
Thanks, Jim. And our next poster is Thinking Systems, Thinking and Decision Making by Utilizing ISS Training Materials. So that's an interesting application of training materials, one I haven't seen before. And that will be by Julia Brodsky, Art of Inquiry, LLC. Please, if you're on the net, please type in your Q's, your questions at the Q&A logo down at the bottom of your screen so we can have some questions as we come out of this 5-minute presentation. Let's go ahead and run the presentation, please.
Teaching systems thinking and decision making by utilizing the International Space Station training materials. I'm a former ISS astronaut instructor and NASA systems engineer, and currently a K-12 educator and education researcher at the Blue Marble Institute of Science. It is easy to see why I'm so interested in systems thinking and so concerned about the lack of this type of education in our schools. Given how fast we change the world and how many complex systems we create, it is extremely important to our students to get introduced to the ideas of mental models, feedback loops, time delays, interconnections, and decision-making in times of uncertainty. Unfortunately, there is not much emphasis on that in schools. I'm trying to address this need by developing courses for informal educators as well as online and summer programs. There are several reasons why I turned to the ISS materials in order to develop a rich curriculum for middle school children, supplemented by the systems thinking materials from the Waters Foundation. For many years, I've been involved with creating, testing, and using the ISS training materials and flight procedures. I understand the complexity of the space station systems, their interactions, and the complicated mishap scenarios. And given my background teaching astronauts, flight controllers, college students, and K-12 students, I can explain these concepts well. The ISS itself provides an amazingly rich and exciting set of materials that is easily adapted to students' needs, providing countless examples of complex structures on different levels, as well as decision-making processes involved in space station design, planning, trade-offs, and operations. The opportunity to interact with these materials provides students with better understanding of the day-to-day challenges of a space engineer, astronaut, or flight controller, helping them make informed career choices. The Art of Inquiry curriculum has been tested both online as a standalone course and in face-to-face settings of traditional schools and summer camps, and was welcomed by students and teachers alike. The students got introduced to the space station through videos, Lego models, and the ISS Familiarization Manual, the original version of which I helped author. The mere idea of using the same training materials as astronauts turns out to be a source of inspiration for the students. The students were asked to role-play NASA directors and flight directors, setting the most important priorities for the ISS from their own perspective. It is notable that one of the objectives suggested was a bigger involvement of underrepresented groups in the space industry, and another one was the search for life in space, as well as space habitat development. The Hazards and Operations board game development is one example of our approach. The students are asked to brainstorm a list of space flight hazards, as well as risk mitigation strategies for each one, and finally develop a board game based on the list. The students put much time, wit, and effort into their projects, analyzing the mishap scenarios that actually took place aboard the ISS and Mir space stations over the years for background knowledge. Later, they used their risk list to propose a better and safer spacecraft design. The course helps students develop an understanding of how little things can cause big problems, how everything on board the space station is interconnected, how difficult it is to predict consequences, how important it is to consider different scenarios and get prepared ahead of time, and how thoughtful decisions need to be made in real time based on knowledge of the system. Students and their families alike find this course to be engaging and valuable for both learning about the space industry and fostering mental resilience and a growth mindset. We hope that we will be able to expand this program to involve more students. So far, we offer several scholarships to low-income kids. If your company is interested in sponsoring some students, or if you are interested in collaborating with us in some other way, please get in touch. And we will continue our research into the best practices of teaching systems thinking with the ISS materials as a foundation.
All right. Well, thank you very much, Julia. And if you would activate your camera now and your microphone, there we go. Ah, there we can see the top of your head. We don't have any questions at this moment. So what we're going to do is let you go ahead and expand on anything you want to on your poster. And then when a question pops in, and a question just did pop in just a moment. As a STEAM informal educator, I appreciate your cross-cultural approach. Are your lesson plans available?
Yes. First of all, I have a blog that I recently posted on Medium that kind of tells a little bit about our lesson plans. But also, of course, I do have some lesson plans available. If you want to contact me offline, I will be happy to share those with you.
All right. That was the one question thus far. Did you do astronaut training as part of USA?
Yes, I worked at USA. It was just at the beginning when the ISS was just starting. So I trained the first 4 expeditions to the ISS.
So is it different training school children versus astronauts? Which is harder?
You know what? People ask me another question. People ask me, what do I prefer more, teaching astronauts and flight controllers or teaching kids? And I would tell you it's more difficult to teach kids, of course. But it's also very intellectually stimulating because the kids are very open. The kids really want to explore new things. And when you give them an opportunity to brainstorm, when you give them an opportunity to look for feedback loops, to look for interconnections, they're so grateful. They do appreciate complexity. This is one of the findings that, you know, some of my observations over the years, that we do not provide them enough complexity, enough exposure to complex situations, enough exposure to situations where they should practice decision making. And they really do appreciate that. This is true for all kids, for kids from different socioeconomic strata. I have kids who come to me from very low-income families. They're as willing to explore these topics as the kids from higher-income families. This type of projects allows the kids to collaborate, to problem solve, and they really, really enjoy it. So it's not easy for the kids to solve these problems. Planning is not a skill that the kids really learn in school, unfortunately. When you have to start from the beginning and plan a project, they don't know how to collaborate. They don't know how to explain their vision to other members of their team. They don't know how to resolve conflicts. So all of that, you know, we practice all of that. It's not just system thinking, as applied to particular technical field, it's also the system thinking as they reflect on how they work within the team.
Okay, very good. And thank you very much for bringing us your poster. I appreciate it, and it must be very satisfying. Now it's time to move to our next poster.
Thank you.
And it is the International Space Station Servicing Robot by Samantha Glasner, Northeastern University. So let's play her slides, please.
Hello, and welcome to the International Space Station Servicing Robot poster. My name is Samantha Glasner, and my teammates include Lamia Farah, Daniel Fisher, William Spencer, Erica Trainey, and our advisor, Dr. Marilyn Minus. This project was produced in the Department of Mechanical Engineering at Northeastern University. Astronaut time aboard the International Space Station, or ISS, is in short supply and is extremely valuable. While most of their time on the ISS is spent contributing to groundbreaking experiments, astronauts must also perform monotonous maintenance tasks. An example of one of these tasks is the routine cleaning of handrails that the astronauts use to traverse the inside of the ISS. This team dedicated our senior capstone research project to designing and prototyping an autonomous robot that will navigate along the internal walls of the ISS to perform an example servicing task of cleaning blue handrails, therefore freeing astronaut time for more scientific experimentation. The main design focus areas were condensed into 1. Cleaning, how the robot will clean the handrails, 2. Navigation, how the robot will traverse the ISS, and 3. Locomotion, how the robot will move along the ISS walls in zero gravity. A design comprised of a four-wheeled robot, a novel soft robotic cleaning mechanism, and an onboard Raspberry Pi controlled simultaneous localization and mapping, or SLAM, and handrail detection system was created. Section 1. Cleaning. A soft robotic cleaning mechanism was designed with a cleaning solution irrigation tubes and vibration motors embedded in the silicone. This soft component was encased in a hard 3D printed housing with clip-in points for the astronauts to install a lint-free dry wipe. The cleaning mechanism was connected to the robot with a single degree of freedom arm. Section 2. Navigation. To allow the robot to see handrails and obstacles while knowing its position in space, the following sensors and cameras were utilized. First, a 2D camera was used to detect handrails. Second, a 2D light detection and ranging, or LiDAR sensor, was used to implement a SLAM algorithm to construct a map of the environment and localize the robot in space. And third, infrared detection sensors and passive infrared motion detection sensors were used to avoid fixed and moving obstacles respectively. Section 3. Locomotion. The locomotion aspect of the robot consisted of how the robot would move through the ISS in addition to how it would adhere to the walls of the ISS in zero gravity. To limit the complexity of navigating certain corners of the ISS and turning, the wheeled robot with tank drive wheels was chosen. The two drive motors were equipped with encoders to feed back into the navigation system. A ducted propeller was mounted to the top of the robot to provide continuous downforce to keep the vehicle adhered to the walls. Section 4. Analytical investigation. To help understand the geometric constraints of the vehicle, the team simulated the robot's ability to navigate the corners and steps of the International Space Station, as well as the ability to drive over obstacles, was confirmed in computer-aided design, or CAD, simulation. The team also simulated vehicle navigation operations using matplotlib Python plotting. Acoustic analysis was employed to determine the vehicle satisfies the ISS environmental requirements for noise. The quality of wetting the wipe, the intensity of vibration, as well as the inflation of the soft robot were guided by simulation methods and empirical data, and were demonstrated using the first version of the soft robot. The accuracy of the camera's handrail detection, as well as the determination of the handrail distance, was tested by doing static tests and doing a 3D printed mock handrail. Section 5. Operations. A program deciphers images taken on the camera and segments out pictures that match the characteristics of a handrail, that it is blue, and that it creates a rectangular shape. Once the robot has identified a handrail, it navigates to the handrail using SLAM to keep track of its position and infrared sensors to avoid obstacles. The cleaning mechanism is deployed over the handrail by rotating the attaching arm. The soft robot is inflated to grip the handrail. A small amount of cleaning solution is applied to the cleaning wipe via irrigation lines cast into the soft robot. The soft robot applies mechanical cleaning via vibration. Once cleaning is complete, the soft robot deflates and the arm rotates up to disengage the handrail. The cleaning mechanism was designed to clean the width of the shortest handrail. For longer handrails, the robot would drive down the side of the rail and then repeat these steps until the entire handrail was clean. Once the robot cleans all handrails on a given plane, it will move to the next plane. This process will be repeated for all the planes of the International Space Station's interior.
So Samantha, thank you very much. This is a voice from the darkness.
Hi, thank you for
Hi.
having me.
We're glad to have you. So we don't have any questions right now, so we'll let you ad-lib for a couple of minutes.
Okay,
Why don't you address what you would like on your presentation?
Absolutely.
That you didn't have time to address in the 5-minute summary.
Sounds good. I actually see something in the Q&A. It says, what benefits
just popped up.
are there, what benefits are there to being attached to the ISS rather than a free flyer? So that's a really great question. There's actually a lot of heritage with free flyers in the International Space Station. Examples are the Spheres robot and now the new Astrobee. And so those are free flyers in the ISS. The Spheres use CO2 canisters to maneuver, and the Astrobee now uses ducted fan system. But we decided to do a wall-mounted version where we used our fan to create a normal force to always push us against the wall. For two reasons. One, just the logistics of us being able to test it on ground. It was a lot simpler to have just this kind of wall-mounted system. And then we could test it using a single air bearing to just give us like a single XY axis of zero gravity effect, but still be able to test our systems feasibly within our capstone senior design project. And then the second reason was that we think that in general, having a mix of heterogeneous robots doing servicing for things like the International Space Station and Gateway is advantageous. So having free flyers is good, but also having robots that maneuver the surface is also advantageous in order to be able to, you get a better grip against whatever surface if you want to touch something on the surface or insert something into a payload bay or do some sort of interaction with the wall of the stations. We thought that having this kind of normal force against the wall would be useful.
Okay, so right now you're just in ground systems activities. Is there any plan to take this to ISS?
So if we were to find some way to do that, we would really love to. In the scope of this, we did it as a kind of final senior design project. So we did it at Northeast University over the last spring in 2020. But we don't currently have any connections with NASA to actually put it up there. But that would be something we'd be very interested in. All of us have graduated and we have someone working at Johnson in mission control now. And I'm doing my PhD at Virginia Tech in in-space assembly robotics. So we're continuing the effort, but there's no plans currently of putting this specific robot in space.
Thank you. So the next question is, expand a little bit about how you made it stick to the wall. Explain your ducted fan a bit.
Yeah, so for the ducted fan, we basically just mounted a fan on top of our robot and then spun it continuously. And we did some testing to determine if the sound from the fan would disrupt astronauts. So we did some acoustics testing and made sure it was below the International Space Station's noise requirements. But in general, we just had to create enough normal force to keep our robot against the wall. And the main force that would be pushing us against the wall was whenever we were traversing over obstacles. So if we had to drive over a handrail or some obstacle, we had designed these soft wheels that we cast out of silicone, and that allowed it to squish a little. So there's a little give, but we basically looked at the maximum force that would be pushing us out from the wall. And then we just made sure that our fan could give us enough thrust to push us into the wall. And so we did some basic free body diagram analysis to figure that out.
Okay, and the final question, any biomimicry aspects of the design? Did you copy any living beings, species, peoples, dogs, cats, geckos, geckos?
Yeah, great question. So when we were doing, we did a lot with this kind of soft robotic idea of cleaning the handrails. And also we used the silicone, which is commonly used for soft robots, in our wheels. So we did look a lot at if we could use different types of biomimicry to actually grip onto the wall. So before we decided to go with the fan system to keep it against the wall, we also looked at gecko grippers, which are research in academia and at NASA Jet Propulsion Laboratory. So that's a very interesting bio-inspired kind of surface to allow you to grab onto surfaces. But we ended up deciding to use our ducted fan, but we did look at some bio-inspired designs.
Well, thank you very much. I really appreciate it. I think this has been great, and it's we can tell you're having a lot of fun with it, and we wish you well as you continue.
Thank you so much.
All right, so it's time to move on to the next opportunity that we have. And what we have there is, all right, the Compact, a proposed ISS-based experiment to study the dynamics and structure of multi-particle systems on the atomic level. From Yui Konopka, an associate professor at Auburn University. Let's play her slide.
I would like to introduce you to Compact, a proposed multinational experiment to study the dynamics and structure of large particle systems resolved down to the spatial and dynamic scales of individual particles, or quasi, a velocity and position measurement of the atoms in the system. COMPACTS stands for Complex Plasma Facility, because a complex plasma will be used as a model system. A complex plasma consists of charged, well-defined microparticles embedded in a bath of plasma and neutrals. The plasma background is responsible for the charging of the microparticles, their interaction, and their dynamic properties. By changing the background, systems can be realized that cover a large dynamic and coupling parameter range, from undamped to overdamped, weakly to strongly coupled. Complex plasmas are naturally optically thin. Any part of the complex plasma volume can be studied by dedicated illumination techniques. Even the interior, many thousands of layers away from the boundaries. However, a complex plasma has one major disadvantage. Particles have to be levitated against gravity. This usually requires the introduction of anisotropic force fields. To avoid this problem, it is preferable to invest in experimental setups that can be operated under freefall conditions instead, for example with COMPACT and the International Space Station. The extreme differences between ground-based complex plasma experiment and those performed aboard the ISS are shown on the left bottom insert. As the timeline in the middle indicates, COMPACT is the most recent development in a series of successfully operated complex plasma experiments aboard the ISS, namely Pika Nevedov, Pika 3 Plus, and Pika 4. The latter actually is still operated aboard the ISS. COMPACT represents a major milestone. Systems with billions of individual particles will finally become accessible, allowing to bridge the gap to continuous media. Also, a new advanced plasma manipulation system promises a new quality of experiments. With COMPACT, an extensive effort has started to incorporate and grow an international interdisciplinary community, expanding beyond the usual complex plasma research groups. The goal is to establish complex plasma as a universal multi-particle model system. If you're interested in COMPACT and its capabilities, you're welcome to join the effort and help to specify and utilize the experiment. Is it really worth the effort? A list of potential topics that might be studied with COMPACT are listed in the top right corner. The list is open for discussion and expansion. Shown in the center of the poster are 3 examples that demonstrate the advanced capability of COMPACT, where large complex plasma systems could be realized even in ground-based laboratories. One example shows the study of wave generation in the low-damped system and the change of the wave features as the complex plasma is compressed. See the top right corner. A second example in the middle shows the simultaneous development of large volumetric vortex flows close to the electrode edges while the center of the system is in a crystalline state. The third example, on the bottom shows the onset of crystallization of a large complex plasma cloud, triggered by the compression of the system and thus the change of the particle coupling strength beyond a certain level that is necessary to start the crystallization process. A full crystallization of the system might take actually several minutes. During this time, the growth of the individual crystallites can be observed and, via 3D scans, studied in full detail. You can extrapolate for yourself how a system under free fall condition in these cases might look like. Please contact me or any of my co-workers if you have questions and/or if you would like to contribute to this multidisciplinary international research project. Thank you.
All right. Thank you. Go ahead and activate your camera and your microphone if you have not already. Yeah. I apologize for the pronoun issue.
No problem at all. Then I have to apologize for my accent, sorry.
Oh, all right. We may start requiring pictures just so I don't screw up like that.
Don't worry.
So as we go forward, we're going to, we don't have any questions yet. When one pops in, we'll jump in and interrupt you and my little dialogue. But what I think that first we'd like to do is give you an opportunity to speak off the cuff for a couple of minutes. Yeah. And expand on anything that you want to in your poster, and we'll jump in and interrupt if we get a question. Otherwise, you have about 4 minutes that you can expand on what you said.
Yeah, it's always weird to hear yourself speak. It sounds a little bit like an advertisement or so, but to some extent it is so, and it's going actually beyond that. We would like to invite people to study our specific system, but maybe also to utilize a potential space experiment and use it in a way which is not foreseen yet. So actually we are just in the process. In the top left you see the program, how it started like in 2000, but with the last one, with the compact one, we actually tried to build a new facility which definitely will be much better. And we try also to open the community to fields like solid state physics, fluid physics, and others, who are actually more the experts than we are in this topic. So we are the expert for the plasma physics background, but not everybody's interested in the plasma, but in the multi-particle system. And also, since those experiments cost a lot of money, it's probably reasonable to consider doing them for a different purpose, not only for dusty plasma, but also like I saw a talk today about liquids, the droplets sticking to surface. So I'm hesitating a little bit to put in liquids in the moment when you try to do complex plasma, because it's a vacuum system, but that could be maybe at a later stage or so. So we really would like to open the system and see what kind of community we could get together to use an experiment of this form to make it really value and useful for many people. So that's kind of one of the interesting things, what the message I wanted to say here. But the examples what I gave here, I hope it's kind of really impressive. Like when I started studying complex plasma, it's like you see physics with your naked eye. The dynamic timescales are in the order of 100 hertz or less. So standard camera systems work for observing things. You just took a laser sheet and you observe any three-dimensional systems and in a cross-sectional detail, or what we would like to do with this experiment, also use 3D optical diagnostics for stereoscopic measurements. And well, I find it always impressive if you can watch a system and how it starts crystallizing, and you see the individual crystallites moving in from different directions, and then you can study how the energy is dissipated in the system and all this kind of stuff. And on the right side, actually, there's one funny thing, the lower picture of this experiment. There's actually, we saw the start of turbulence in the curved flow, and you see it on the scale of the atomic level. But we are not the expert to analyze that, so we really would need more input from different communities who are more expert and on thermodynamics and so on.
All right, well, thank you very much. I appreciate you being able to expound off the cuff like that. You certainly understand this, and you certainly are having a good time.
Yeah, I understand the plasma part, but I really need experts on the other parts.
Need that help. All right, great. Well, thank you very much. It's time to move on, and I'm going to be handing the reins over to my co-MC here, Sheng-wey.
Excellent. Thank you very much, Alan. All right. Let's see. Our next one has another fun title here. Our next one is called May the Force Be With Mechanosensitive Proteins and Bone Remodeling Cells in Microgravity. Fane Antoine from the Florida Gulf Coast University is our author for this one. So let's roll that poster, please.
Hello, my name is Fane, and our second presenter will be Sabrina, and today we are going to discuss mechanosensitive protein and bone tissue. We know gravitational forces on Earth are essential in physiology, and microgravity has a significant effect on physiology, especially in the muscular and skeletal system. In microgravity, muscle atrophy and bone density loss occur, but we don't know the detailed mechanism involved in those processes. For our research, we look at the transient receptor potential ion channels, or otherwise known as the TRP proteins, since these are mechanosensitive proteins that have been shown on Earth to be associated with bone remodeling. There are different types of TRP proteins, and the ones we're going to work on, it's the TRPV1 and TRPV4. TRPV1 proteins are associated with bone formation by osteoblasts, and TRPV4 proteins are associated with bone absorption by osteoclasts. We wanted to test the hypothesis that TRP proteins distribution differ between Earth and spaceflight specimens. To do so, we use immunohistochemistry and histology on mice from Rodent Research Reference Mission 1 that were maintained at the space station and habitat control on the ground.
TRPV1 and TRPV4 labeling was visible in the periodontal ligament of the habitat ground control in spaceflight mouse mandibles. I'm going to see in this image right here where I'm pointing to is the periodontal ligament, and this is the tooth right here, and then to the right we also have another periodontal ligament surrounding the tooth. If we go right down here to this graph, we can see that the periodontal ligament was significantly wider in the spaceflight specimens compared to the ground control. So on the x-axis we have the ground control right here and the spaceflight right here. And on the y is the mean PDL of the width. And we can see that the spaceflight had more than the ground control. As we go to the right, this graph, we're looking at the TRPV1 proteins and TRPV4 proteins on the x-axis and the mean intensity on the y-axis. We see that the TRPV1 labeling was significantly more intense in the spaceflight specimens compared to the habitat ground control specimens. And in the TRPV4, to the right here, we can see there was no difference was observed in the labeling. As we go right down here, this is an H&E stain. Right here is the alveolar bone covered by the periodontal ligaments, and then right here is the tooth. So the graph to the right of it, we have the ground control and the spaceflight on the x-axis, and the y-axis we look at the mean number of osteocytes that we have collected. We can see here that the mean number of osteocytes was significantly greater in the spaceflight specimens compared to the habitat ground control. Okay, awesome. Okay. Dun, dun, dun. So we know that microgravity influences homeostasis, and that is imperative to predict the physiological changes to prepare for humans to go into space travel, and also to assure that there's proper functioning while in space, and to maximize recovery upon return to Earth. Mechanosensors such as TRP proteins may have the capacity to detect gravitational forces, changes that drive bone remodeling activities observed in astronauts and animals that experience microgravity. The wider periodontal ligament in spaceflight specimens was observed here, in contrast to other data that we've also seen, that tissue swelling is associated with fluid and pressure shifts in microgravity. TRPV1 and TRPV4 labeling was localized in the PDL and the tooth pulp. The PDL may receive numerous sensory afferents known for high TRP expressions, TRP expressions. We also saw the greater number of osteocytes of intensity in the TRPV1 labeling observed in the spaceflight specimens is consistent with the theories that TRPV1 plays a role in bone formation. This data suggests mandible bone remodeling may differ from that of long bones, which exhibits bone density reduction in microgravity. This data provides insight into the possible differential regulation of bone homeostasis in tissues and in space conditions and on Earth.
We want to
Okay, so that's the end of that poster, I guess. So Fane, bring yourself up on screen. Yeah, that's the, there we go. And once Fane comes up, we'll please, everybody out there in the webosphere, please feel free to submit some questions for these guys. It's a pretty interesting project. Fane is working on it. There they are. And wearing your masks. Excellent. Good on you. So we don't have any questions right now, but I'll ask one quick question. So based on your work, what would you do differently next time if you had another similar opportunity?
I don't think we would change anything.
It wouldn't change anything per se, but maybe more time to to continue this research and analyze the data, because I think we got the specimens in February, and then the pandemic happened, so we didn't have a lot of time to cut the tissue, process them, and then just figure out exactly what else is involved with that.
Gotcha. Yeah, lab work took a big hit during this pandemic. Yes, absolutely. And so we do have a submitted question. Did you look at what TRPVs initiated as signaling in tested conditions? Oh, you guys can see that on screen? Okay. Do you understand the question?
No.
Okay.
We did not test that, and we didn't have the time to analyze it.
Okay. Well, until there's another question, you have about 3 minutes to talk. Oh, there's another one, sorry. Are astronaut mandibular stressors, given their diets on ISS, different? So I guess the difference between rodents and astronauts. Did you get a chance to look at that?
For rodents, because they grind their teeth, and I mean they have to in order to maintain like the length of their teeth. We think that let's see. I don't think there's much difference.
Okay. Okay. It's something that you could look into, maybe. Why did you choose the TRP biomarker for bone remodeling? Can you guys hear me okay?
Yeah, we're just trying to see the question.
Oh, okay.
Why do you choose a trip biomarker for bone modeling? For the labeling, I guess, right? To see.
Be sure to speak up into the camera.
So we use a trip biomarker for the bone modeling just to label everything, see what we're looking at exactly. If that's what you guys mean by the question.
Okay. Yeah. And then one last one, I guess. What do you know about the space mice diet versus the land-based ground control mice? Was there any difference there? Was the diet strictly controlled to be identical for your ground-based versus your space-based specimens?
We don't know much about the space mice diet, whether from ground and the space flight specimens. We just got the samples, and
okay.
We just got the samples and we just analyzed. We got young and old mandibles from our from space and control. Yeah, we don't know anything about the diet per se.
Okay, gotcha. And then just one last quick question. So this is a clarification on the one before. So a human in space, do they chew any differently than they do on Earth? You know, do they chew harder? Do they chew softer? I'm not sure if that's within the scope of your project here.
It's definitely something we can look into because right now we're graduating this semester. So
congratulations.
Thank you. Future students can continue to work on that and we can find out more.
All right. Well, very good. Thank you so much for your time, you guys. That was great. Really interesting stuff. And we're gonna, thanks. Okay, we're gonna get started with our next poster here. The title of this one is Understanding Manufacturing Techniques for the Development of the Sustainable Commercial Case for the International Space Station. Our presenter is Douglas Marsh from Martian Sky Industries, LLC. Go with the poster.
I'm Doug Marsh, the founder and CEO of Martian Sky Industries. Today I'm pleased to be discussing the topic, Understanding Manufacturing Techniques for the Development of the Sustainable Commercial Case for the International Space Station. As the ISS transitions toward a more commercialized entity, it is important for manufacturers to understand how the development of useful techniques impacts the commercial case and provides the foundation for the advancement of humanity in the space market. We'll start the discussion by defining commercialization so we can begin to formulate the idea for how space manufacturing techniques impact the commercial case. Commercialization is the process of managing or running something principally for financial gain. In manufacturing, this occurs through creating a higher quality product, reducing the cost to the customer, or shrinking the timeline to get the component to the end user. More importantly, the development of the sustainable commercial case in space follows the same path as ground-based manufacturing operations. So, just as the Industrial Revolution improved human mobility and interaction on Earth, the same is primed to occur in space. We must recognize that as we approach new worlds with new opportunities, we are approaching them from space to ground, which is the reverse of how we're used to thinking about development tracks. The more infrastructure that manufacturing provides from in-space operations, the faster the timeline to colonization efforts or quick science missions that we can provide. Thus far, significant emphasis has been placed on additive manufacturing techniques in space. These certainly stand to provide great value for deep space missions and reusability of resources, but commercial sustainability requires development across all manufacturing processes to drive proper market maturity to gain the best advantage for the customer. We must also begin to move beyond just product achievement and develop a true commercial market that focuses on process enhancements to drive necessary growth and competition in the market. Resources must be allocated to develop standard manufacturing techniques now, and I stress now, so that processes are streamlined as commercialization takes over. The ability to push this envelope now requires heavy investment in automation. Let's unpack the role of automation in space manufacturing. Automation is a key driver in meeting process enhancement goals. The ability to formulate self-reliant technologies allows the manufacturing techniques to do all the work in the background with minimal human interaction. This creates what I call a celestial terrestrial value case, where the automated method provides an equal value to ground-based operations and further understanding of operational pain points are expanded through the use on Earth. This also acts to reduce reliance on ground operations and manufacturing processes, allowing the technology to push the sphere of influence further and further away from the surface of the Earth. We need to understand what the growth looks like internally for manufacturers. Well, as the understanding of manufacturing processes increases on orbit, new products begin to create new markets, a service sector to support these markets comes into place, quality and affordability of life in space improves, and better products become available. This creates a number of niche markets for manufacturers to operate in with growth in production, logistics, marketing, sales, and support teams. The manufacturing and space sectors are in position for symbiotic growth that allows for current knowledge bases to transition between them. This is relying on us pushing for better scientific data and answers to manufacturing questions now, so we can see that realized expansion. By focusing on how all manufacturing methods can be used to create better or more affordable products, or how different techniques stand to reduce the timeline, we can truly begin to develop the commercial case aboard the ISS. This concludes my presentation. Please feel free to reach out to me at doug@martianskyindustries.com if you have any questions or comments regarding the presentation. Thank you to the ISSR&D conference for allowing me this opportunity, and let's do great things.
All right, very good. Thank you, Doug. So Doug, bring yourself up on screen here so we can talk a little bit, and hopefully some questions will come in through the chat. There you are. Good to see you, Doug.
Hello.
Thank you for that. So we don't have any questions right away, so why don't you feel free to talk a little bit more about your presentation and your thoughts.
Yeah, as we go more to a commercial-based low Earth orbit economy, we really need to start looking at how we can develop greater processes and begin to look at processes that we might even assume will be easy to transition, because we don't want to come to find that something that we believe is going to be easy to accomplish in space is really just laden in a number of problems that we hadn't previously assumed. So as we're looking at space and the market in space, we need to look at what can drive costs down for the customers. We need to look at it as I have here, the cost, time, quality aspect of things. And the full and honest reality is, when we're trying to develop a commercial manufacturing process that improves what we can gain on Earth, we understand that cost and time really aren't something that we're going to be comparable with against industry on the ground. And so what we're really focusing on is development of quality in products. And so is there a way that we can develop a product that has an improvement and extends your lifetime, whatever it may be, from what we can produce on Earth?
Great, Doug, thank you. So we do have a question. So what is your estimate of ROI on space startups by maybe 1 or 2 years? I mean, that's a really short time frame. Is it possible to look at an ROI right now?
I would say that's awfully soon. When we're developing in space, I think 5 years, 10 years is well worth planning for. 1 to 2 years, you're really pushing development. It would depend on the size of your team. So, if you're pushing a large team of folks, yeah, you can start pushing a lot of technology out over the course of 1 year to 2 years. But if your team's small, yeah, it's going to take a long time to sort of build that snowball that's rolling down the hill before it really gains the momentum you're looking for.
Yeah, and and I totally get your point about the whole cost time quality balance, and cost and time are not not something that you're going to do better than ground-based systems off the ground. So thinking about the quality of things, what kind of products do you think fit into this category of being a possible candidate to get that quality that you need without being able to compete on cost or time?
Yeah, and to that, we really have to look at heavier into crystal formations are heavily studied. There's a number of things that are already fibers, obviously, something that's received heavy amounts of study, but I think there's a lot of look into liquid formations, resins, things like that, that really still are pretty open for what can be developed on the ISS and don't have a lot of background in science at this point. So,
gotcha. Okay, and I think that answered the question that popped up while I was actually asking it, which is what industries do you think would be kind of the low-hanging fruits? You know, so the easy wins, but like you said, crystals, fibers, things like that, right?
Yeah, glass manufacturing, you know, things of that nature certainly are the low-hanging fruits and are already being pursued. So, you know, there'll be levels of things that we can, you know, run into and you know, try and test and manufacture. But, you know, I do want to say, you know, the low-hanging fruit is important, but everything's important if we're developing the true commercial case. So we need to look at all processes and how we can transition.
Very good. Excellent. Well, we're out of time. Doug, thank you so much. Appreciate your being here. And
thank you.
Yeah, and his information's on the slide there if anybody else wants to reach out. All right, so moving on. Our next poster is the Molecular Muscle Experiment 2. This time it's back and it's angry. Molecular mechanisms and countermeasures to muscle decline in space. Timothy Etheridge from the University of Exeter is our presenter. Let's roll the poster and its narration. Stand by one, we'll get this poster up in a second.
Hello, I'm going to be talking through my poster entitled The Molecular Muscle Experiment 2, or MME2 for short, Molecular Mechanisms and Countermeasures to Muscle Decline in Space. The heritage to this MME2 experiment is that we've been using the tiny worm C. elegans as a model for space biology for many years. For several practical advantages, they're small, relatively easy to culture, cheap, and they require very little upmass compared to some other biological models. But importantly, that in microgravity, worms display very similar gross molecular changes as to all other species tested to date, including astronauts. The two of these include changes in cell adhesion signaling and also insulin signaling. And actually, part of the heritage of MME2 is our first MME experiment, where we aim to probe these two pathways, cell adhesion and insulin signaling, as mechanistic regulators of spaceflight maladaptation, but also as therapeutic targets. So we flew genetic and pharmacologic interventions to try and counter these abnormal changes in flight and see whether we could prevent some of the health decline. The analysis of these samples is still ongoing, but early results indicate DAF16 or FOXO activation may be a primary mechanism of spaceflight changes that could account for a lot of the metabolic changes that occur in flight. So figure 1 here shows DAF16 GFP translocation to the nucleus, suggestive of DAF16 transcription factor activation. If we move to figure 2, we've also found so far reduced levels of the neurotransmitter dopamine in spaceflight samples, which could also account for some of the neuromuscular decline that we see in flight. And thirdly, our collaborators in the United States with Monica Driscoll has found abnormal neuronal protein garbage extrusion and handling, such that we see greater amounts of these neuronal protein aggregates occurring outside of the neuronal cells themselves in the hypodermis, which is abnormal and not seen in ground control samples very much. So we think that figures 2 and 3 combined, and more data suggest abnormal neuronal function in the spaceflight environment. Moving on to the Molecular Muscle 2 experiment, we are taking ongoing data from various species, including worms, but also rodent and astronauts, to probe further mechanisms of spaceflight changes. Here we're going to be focusing on the role of neuromuscular signaling as a mechanism of flight-induced muscle decline. We'll be probing the role of altered calcium handling, and we'll also be looking at the role of altered mitochondrial function, and again, flying some drug interventions to try and counter these changes. Interestingly, this will be the first UK national payload on board the International Space Station. But it's not a UK-only mission. We've brought in an international team, including Japan, China, South Korea, the United States, and GeneLab, all with their own hypotheses and their own roles to play to look at those mechanisms and countermeasures to flight-induced health decline. Our flight operations are, by design, pretty simple. We're essentially just going to grow worms in 6.5 milliliters of liquid bacterial feed inside gas permeable bags for gas exchange. And we have done science verification testing from MME1 that shows that these worms can grow in this environment. The experiment hardware, the ISSA ECs, which house 3 culture bags per experimental cassette, of which will fly 24 for a total of 72 bag flight conditions. Importantly, because we're working with Kaiser Space, a UK-based flight provider, commercial access to the International Space Station is now available from the UK, and Kaiser Space can be contacted for anyone interested in this. The flight operations themselves are also relatively straightforward. We'll launch worms with passive cooling, phase change material, and dock with the ISS. They'll be placed into cubic at 20 degrees centigrade for about 5.5 days to grow to young adults of the first generation, and then placed at minus 80 degrees C in Melfi to stop the experiment before returning to Earth. Anyone that's interested and would like to find out more can go to Kaiser Space website and contact them. Of course, look at the UK Space Agency and ESA websites, or we have our own MME dedicated website that people can refer to for more information and some outreach materials that are being updated over the next 6 months also, or you can contact us directly at the MME team. Thank you very much for listening. Goodbye.
Okay. Thank you, Tim. Very interesting project there, very interesting experiment. We don't have any current questions posted right now, so you've got about 3 minutes or so to talk freely about it, and I'll jump in if I have any questions.
Okay. Yeah, so really this most recent MME2 is part of just a bigger program of research that's been going on way before my time for about 20 years now with Nate Shevchuk, who's just moved to Ohio University from the UK. Where I've been using worms to characterize the molecular changes to spaceflight environment for a long time. What really MME1 and now MME2 doing are exploiting that transcriptional information that we know what genes are changing in flight to now fly the targeted interventions to try and see if we can stop those negative changes happening in flight. The nice thing about MME2 is that it's the first UK national payload, as I said in the slide. So we've got this capacity now to implement and fly spaceflight ISS experiments from the UK now, but with the appreciation that it's always better to bring in other people. So it's an international team of researchers, as I say, so it's great to have all these different countries involved to explore their own hypotheses of interest. I will say that associated with this project, kind of briefly mentioned it at the end there, is that there's a big outreach aspect to this whole project. So we're really keen to bring schools into the fold as much as possible. We've reached tens of thousands of members of the public so far with our targeted outreach activities. Some of those activities are available on our MME website. So if anyone's interested in any of the school-based activities that we do, it'd be great to hear from them.
Excellent. That's great. We did just get a question for you. How many worms can your studies use?
So we fly them in these little bags. ESA very kindly gave us way more conditions than we originally planned for in our first flight. So we fly about 72 conditions, so 72 bags, and each bag, depending on the researchers and their experimental design, we fly anywhere between 5 to 5,000 worms, and with progeny we return about 40,000 animals. So loads to get enough biomass to do the genetic analysis we need.
Wow, that's amazing. And congratulations on being the UK's first payload. That's fantastic.
Thank you.
Yeah, that's great. And so you're doing a lot of outreach to the schools and everything about this, and talking with them about this project?
Yeah, so it's integral to us. We're really keen on this aspect of the project and exploit that fascination of the public with flight. So we designed specific outreach activities that we travel around the country doing. We get the public hands-on with the hardware. We explain to them. We have all these posters printed out so we can describe to them. We have videos. But we also, on our webpage, we have school-specific activities that school classes can actually do in their classes.
She misspelled her last name.
Excellent. All right, Timothy, thank you so much. We're out of time.
Thank you.
That was great. People should feel free to contact Tim after the fact and reach out with any questions. Thanks again. I will now turn it over to Alan to introduce our next poster.
All right, next poster time. Move my chair back to where you can see me. I'm trying to hide. So next we've got, these folks have been involved in the ISSR&D conference for a number of years in different levels of presentations and posters. So the first one is Aquior's Aquior's antimicrobial biofilm inhibitors for the environmental control and life support water recovery systems in space. And there'll be Cynthia Buzzle, the founder and CEO of Aquior, who will present to us. Let's run the slide.
I'm Cynthia Brazel, co-founder and CSO of Aquior. Aquior is a biotech company with green antimicrobial treatments. Bacterial contamination is a problem on the International Space Station, especially in the water supply. As an alternative to sending up water to the ISS, there's a water recovery system that recycles wastewater, urine, soapy water, condensation into potable water. This system is critical for life support and getting contaminated by common waterborne bacteria. The current treatments are iodine or silver because of their safety profile, but they're not working. One reason they're not working is because one of the bacterial contaminants in the water is resistant to silver. Another big problem for this water system is biofilm. Biofilm is the attachment of bacteria to surfaces and the production of slime. You can see biofilm slime in a pipe in one of the pictures on the bottom left. Silver and iodine may be able to kill the bacteria floating in the water, but they're unable to kill bacteria in biofilm. Biofilm can degrade materials and clog pipes and increases pathogenicity of the bacteria, which is threatening the health of astronauts. I'm a marine and medical microbiologist, and I discovered natural antimicrobials from the ocean. Aquior now has a portfolio of molecules. Over 25 of them are certified as green and non-toxic. They have broad spectrum antimicrobial and anti-biofilm activity, and they were used to develop a water treatment that was tested by NASA at the Marshall Space Flight Center. Phase 1 of testing screened 8 of Aquior's treatments for antimicrobial activity against mixed species of 3 bacteria found in the water recovery system that are biofilm formers and the silver-resistant strain. After the initial screening, the most effective treatment, A1026, was chosen for Phase 2 testing. Phase 2 testing was done in 55-liter tanks, seen in Figure 1, that simulate the flow of the water on the ISS. Aquior's treatment, A1026, at 0.06%, eliminated the contamination 99.999% of 5-log reduction in the first day and maintained activity for over 15 months. Monitoring determined that the concentration of the treatment did not decrease over time. Biofilm testing was performed by staining the biofilm green and was used to determine the concentration needed to treat the mixed species bacterial biofilm. You can see in Figure 2 that the control A, without treatment, formed thick biofilm in bright green, and Aquior's treatment C at 0.007% prevented biofilm formation, and dead cells can be observed in red. In conclusion, an antimicrobial treatment for the ISS is critical for long-duration manned space travel. Aquior's treatment, A1026, eliminated the contamination and maintained activity for over a year in this closed system. The same treatment can be used on Earth to treat water systems in consumer, industrial, health, and defense sectors. Thank you.
And thank you, Cynthia. If you can activate your camera and your microphone, we'll get right into it.
Hi.
Hi there. Now I want to apologize that we misspelled your name in our system and it took
Oh, no
get
problem.
you promoted into the conference here.
Yeah,
And
no problem.
we don't have any questions yet, so well, we just popped one in. Let's see what it is.
Oh, I got one.
It
Yeah.
says, have you tested these antimicrobials for medical applications, cystic fibrosis, skin wounds, etc., whether in vitro or in animal models? If so, what bacteria were targeted and what was the outcome?
Yeah, great question. We have looked at medical applications, especially for cystic fibrosis, that was using a Pseudomonas aeruginosa mucoid strain because that bacteria specifically forms a biofilm and complicates those infections. Yeah, so with skin wounds also are associated with biofilm. That's basically on the medical side, when biofilm happens in our bodies, antibiotics are unable to treat those infections, and that's why they're associated with chronic or incurable infections. So we haven't gone through FDA, but the NIH and the DoD are interested in helping us with our preclinical trials. So that would be the next step for that.
All right, another question is, what toxicity assays were performed on treated water to validate their safe use in potable generated on board the ISS?
Yeah, so I mentioned we have a portfolio of molecules. The ones that we tested with NASA have gone through EPA TSCA, so they are approved and for this type of use. So specifically in this system, before the astronauts drink the water, it is filtered, and that filtration removes our treatment. But they are considered non-toxic and green for a pre-treatment for water.
Okay. Now, you mentioned that the NASA system's not working. Those are pretty strong words. When do things become critical, and what is NASA doing about that?
Right, so this really becomes a problem for long duration manned space travel, going to Mars, for example, because there is no alternative water. You know, they can't ship it up. So that's why they're really looking into this system and very concerned with the biofilm contamination. Our next step is we have a proposal to do experiments on the ISS. So either it needs to be, our water treatment needs to be tested in microgravity on Earth or microgravity on the ISS.
So is that proposal with ISS National Labs, or are you working directly with NASA on that?
I didn't hear you.
Is that proposal working through the ISS National Labs, or are you working directly with NASA on that proposal to go to the ISS?
Oh, that is a CASIS proposal.
Okay, and finally, the last question from me, unless something else pops up, and that is, are you engaged in any of the activity trying to do planning for the lunar missions and the long-term settlement on the moon?
We are not directly involved in that, but of course, you know, we are an option if they want to do water treatment. So we would be happy to be involved, but we're not yet.
Well, I'm sure they want to do, but then lots of, as in everything else, there's lots of options and lots of ways of going forward. So congratulations on getting as far as you have, and I wish you well as you continue your development work in this antimicrobial world. It's certainly becoming interesting in Earth and in space. So
Yes.
thank you.
Thank you.
And now move on to our next presentation, Cygnus, a platform for technology development and demonstration in low Earth orbit. Our presenter on this will be Robert Thompson from Northrop Grumman Corporation. Run the slide, please.
Hello. This poster presentation is entitled Cygnus, a platform for technology development and demonstration in low Earth orbit. My name is Rob McKnight, and this poster was developed by myself, Tony Foti, and Bob Thompson, and we are members of the Cygnus team at Northrop Grumman in Dulles, Virginia. The purpose of this presentation is to illustrate the ways in which Cygnus has enhanced its capabilities and services to NASA and commercial space as a platform for development and demonstration of new technologies in an operational environment. I will be giving a history of the Cygnus spacecraft, reviewing the updates made to Cygnus over its history, and discussing the ways we have expanded science and commercial space utilization of the vehicle. Cygnus takes cargo to the ISS and removes trash, but we do a whole lot more. The Cygnus spacecraft is made up of two modules. The service module consists of a quad string of avionics, guidance, navigation, and control sensors, redundant communication equipment, along with the propulsion and power subsystems. The pressurized cargo module accommodates pressurized cargo, supplies, and powered experiments, and comes in standard, enhanced, and stretched variants. The Northrop Grumman Cygnus spacecraft was born with the award of the Commercial Orbital Transportation Services Space Act Agreement on February 19, 2008. The Cygnus spacecraft first flew the Orb-D1 mission in 2013 and has now completed 10 missions under the Cargo Resupply Services contract and 2 missions under the CRS-2 contract as of September 2020. To date, Cygnus has delivered over 36,000 kilograms of cargo and disposed of over 27,000 kilograms of trash from the station. Cygnus has accumulated approximately 3 years of on-orbit time with over 748 days berthed to the ISS, with one spacecraft on orbit for over 400 consecutive days. Cygnus has deployed 58 CubeSats from 2 different deployer systems and has operated 9 free-flight hosted payloads, including the Sapphire Fire experiment. Cygnus has launched on 2 different rockets and is the only U.S. vehicle to reboost the ISS since the Space Shuttle retirement. The Cygnus Performance Enhancement Program began with the Orb-2 mission and culminated with a complete set of upgrades on the OA-4 mission, including an enhanced PCM, high-efficiency lightweight solar arrays, and a significant improvement to the Ascent cargo upmass. Beginning with OA-9, Cygnus became the first visiting vehicle to use the upgraded C2V2 space-to-space communication system, paving the way for future vehicles, including commercial crew. With the CRS-2 contract, Cygnus has again increased its cargo upmass and has also added 2 PFRAM attachment points to dispose of up to 2 orbital replacement units up to 1,200 kilograms from space station. In addition, the CRS-2 Cygnus has increased the support for powered mid-deck lockers up to 6 and increased the number of power and data services to support secondary payloads. MDLs and other payloads can now utilize remote access for direct communication between NASA payload providers and their payloads while Cygnus is in orbit, and Cygnus connects NASA payloads directly to the ISS Ethernet network while berthed. Cygnus now supports a cargo manifest of up to 3,729 kilograms of supplies to the International Space Station, including large and non-standard cargo items. 20% of that cargo is handed over and loaded 24 hours prior to launch to provide the maximum flexibility to NASA. This provides delivery to the ISS in less than 96 hours after turnover. In the event of a launch scrub, Cygnus and Antares are able to support refreshing time-sensitive cargo in time for another launch attempt in less than 24 hours. Northrop Grumman provides several science facility options for principal investigators working in multiple areas, including live animals, plant and cell studies, bacterial science, vaccines, human performance, crystal growth and material science, and space and earth science. Facilities include network connections to NASA centers, including Marshall Space Flight Center, along with staff to support operations, coordinate logistics, and support experiment monitoring during missions. Cygnus began adding secondary payload objectives beginning with the OA-4 mission in 2016. Secondary payloads to date have included CubeSat deployers, as mentioned, microgravity research, technology and sensor demonstrations, and re-entry data recorders. The Cygnus architecture provides payload commanding and real-time high-speed data downlink or onboard data recording. A surge capacity for downlink is available using additional commercial ground stations if necessary. The Northrop Grumman Control Center operators work with payload providers to provide services including dynamic vehicle operations such as orbit raising to 450 kilometers, attitude changes, or changes to the vehicle configuration. In conclusion, Cygnus has enhanced its capabilities and services to NASA, as well as to the science community and commercial space industry as a platform for development and demonstration of new technologies and capabilities in low Earth orbit.
All right, thank you very much on that. And Robert, if you would activate your camera and your microphone, please. We'll go ahead and move forward. And I would remind our audience to get the questions in on the Q&A so that we can get some things from you to Northrop Grumman and what they're doing in this. So Robert, we don't have any questions yet. I'll jump in and interrupt you when we do have some. So my first question though is there anything that you would like to expand upon over the next few minutes as folks are bringing their questions? And like I said, I will interrupt you if some questions come in.
Sure. So just to stress some of the key items that were covered in the narration, Cygnus really has, over the last 7 years of its operation of missions delivering cargo to the ISS, enhanced its capabilities, and we continue to do that to serve NASA as our primary customer, as well as the science community of scientific payload developers, as well as engineering developments demonstrating new technologies in space. So Cygnus really has become, over the last 14 missions, a platform that payload and science developers can put their payloads onto Cygnus to demonstrate technologies or demonstrate scientific experiments. The Sapphire Fire experiment that began flying under the CRS-1 contract and continues on the CRS-2 contract is an example of that. Testing, and that's produced by Glenn Research Center, testing different material responses to fire conditions in zero gravity. Obviously, that kind of experiment, due to safety concerns, is difficult to conduct on the ISS itself. So Cygnus, being a payload that goes to ISS and then departs, provides a good way to conduct that kind of experiment. We are also a platform where we can deploy CubeSats through our work with NanoRacks and IOPS, who both fly their payload
I think we lost you for a moment, Robert. There you are, you're back. Can you hear me, Robert?
Yes, I'm here. Sorry about that.
Okay.
So hopefully you can hear me again.
Yes, we can.
So CubeSats can be deployed from Cygnus after our mission to ISS, as well as a number of other secondary payloads that can be attached to the exterior of the Cygnus service module on panel 1. We can provide power data services to a number of different types of payloads. We've posted software-defined radios, various different sensors, and as well as a camera system. So, in conclusion, Cygnus really is a platform that a multitude of different payload developers and scientific experiments can develop their system and fly on Cygnus. And there are actually a number of different mechanisms that could happen. We've developed internal Northrop Grumman payloads that have flown on Cygnus. Other providers have worked through NASA to fly their experiments on Cygnus. And we've also had direct contracts with Northrop Grumman to fly on Cygnus. So there's a variety of different ways to access the spacecraft. And we fly about 2 times a year. We're getting ready to launch our 14th CRS mission here September 29th, which is next Tuesday. Launch is at 10:27 p.m. Eastern time. So we hope everybody will be watching.
All right. So you talked about Cygnus using, having secondary payloads for the ISS missions. When does the ISS mission become secondary because you have enough of your own payloads to pay the bills?
Yeah. There's a lot of discussion on that. So some of our secondary payloads get manifested through the ISS National Lab. And so then that becomes a discussion with the ISS program on, with ISS National Lab on allocating cargo mass to those secondary payloads. And so in that sense, in that case, the secondary payload is part of the primary mission of providing cargo to ISS. So from our spacecraft performance perspective, a mass allocation, a power allocation, and then other resources like mission operators and communication assets are allocated to that secondary mission. So in a sense, the secondary payloads through that channel can be part of the primary mission. The things that we do, if they are Northrop Grumman, Northrop Grumman developed mission that's flying on Cygnus, we generally work with the ISS program so that they understand what we're trying to do. And those Northrop Grumman missions have gone through the ISS National Lab process.
All right, Robert, thank you very much. It's,
thank you.
Really quite an interesting expansion from the old COTS to CRS-1 to CRS-2 programs. And as you're expanding, it's really interesting. So thank you for bringing it to us.
Sure, thank you, Alan.
And now it's time to move to our next experience. We're going to talk about composite coatings for passive spacecraft charge mitigation. And they'll be brought by Timothy Hall, the Principal Research Scientist, Faraday Technology Incorporated. So play his slide, please. Okay, so we have a handful of presentations that do not have audio. So what we're going to do here is we're going to give you guys about 3 minutes, maybe 2 more minutes till 24 after the hour to go ahead and read the slide, and then we'll bring in the presenter to give commentary. Okay, Timothy Hall is. Okay, we found the audio issue and we are bringing the audio up.
Who develop electrochemical processes and techniques for a wide variety of applications. In this case, we're using our unique base, our electrochemical base, to electrophoretically deposit graphene materials onto spacecraft materials like 6061 aluminum, and also using an electrodeposition approach to apply decorations to these graphene materials to further enhance the passive emissivity potential of these surfaces. Specifically within the program, we optimized and developed the process to apply the graphene materials to the surface, as shown on the left. In the middle, we optimized the process to decorate these graphene materials with transition metals like cobalt, nickel, or iron, alkaline earth metals like calcium, magnesium, or barium, or alkaline metals like sodium, lithium, cesium, or rubidium. Then we performed yield measurements at Utah State University, led up by J.R. Dennison's group, where they specifically analyzed the peak emission, the Emax, and also the total range of crossover emissions from the initial onset of greater than 1 to when the range was less than 1. The bottom figure shows the yield curves for bare graphene, the transition metal decorated graphene, alkaline earth metal decorated graphene, and alkali metal graphene. While the top of the far right column discusses the total electron yield, specifically looking at the Emax, the range of crossovers where you're greater than 1, and compares each of these fundamental areas. What we see overall when you compare these is that graphene alone improves the Emax by 1.2%, 1.2 times, while the change of crossover by 1.5. The addition of transition metals, like cobalt, nickel, or iron, improves it almost 2 times with a 4 times increase in the crossover energy. The alkaline earth metals decorated graphene increases the Emax by about 2 times with a 1.5 time increase in the crossover, while the alkali metals decorated graphene improve an Emax by 2.5 times and about 9 times the range of crossover energy. So by applying this coating selectively to spacecraft materials, you have the potential to selectively mitigate charging passively from a satellite's or a solar cell surface. Furthermore, we demonstrated the potential of applying these graphene coatings and achieving transmissiveness of the coating, as shown in the middle of the right column, where if you tailor the thickness of the deposit, you can inherently achieve a transmissive coating, and potentially enabling you to coat the top of solar cells with a passively emitting coating, or selectively coating around the edges where the current electron sinks are used on solar cells that lead to the passive emitters. This work was funded by the U.S. Air Force. If you have any questions or would like to discuss this work in any detail, please feel free to reach out to us at Faraday Technology. The PI on this work is Danny Liu. His email is given at the bottom right. Feel free to reach out to us or anybody at Faraday. We would be happy to discuss our progress with this technology.
All right, Timothy. Thank you for that briefing, and I'm sorry we had problems getting it started. If you'd activate your microphone and camera again, we'll get going. There we are. Good to see you, sir.
You're hard to hear, so.
Okay, I will become more voluminous in more ways than one. So let's talk a little bit about your poster. Do you have anything you want to add to what you talked about in your presentation while we wait for some questions to come in?
I think the biggest thing, we are electrochemists. We are not satellite makers. We are not, we don't know a lot about static emission or passive emission. We're electrochemists, and we're looking for partners to work with to transition this technology. And that's one of the biggest things that I forgot to add into this poster. We're looking for partners to work with that know what devices really need these type of materials.
All right. And so with that, maybe some of my questions might be the wrong questions. So if they are, you just tell me that's what a partner is going to help you do. I wonder if there's any correlation between charge on the spacecraft and magnetic issues and magnetic interference with spacecraft systems.
That's an interesting question. Professor Dennison is our expert physicist on this program. So all of the emission yield measurements that were done by his group. He specifically talked about that Emax, where the Emax is the maximum number of electrons emitted. And whenever it's greater than 1, that means for every electron bombardment, you have the potential of emitting more than that 1 electron. From an electrochemist angle, he also mentioned the range of incident energies, which is that change in crossover, is the broader that is means the higher field you can live with when you're being bombarded with electrons, like traveling through the ionosphere.
Well, you know, there's a long history of aircraft with charges and charging on the skins, and went through a lot of learning on that in the early aircraft days, especially when we got into high-speed aircraft and high-altitude aircraft. And also in submarines, there's been some interesting work having to be done with charge and magnetism. So that may be some place where you can go and get some further information on how to do some of this. Now, did you talk about doing this coating on the interior to reduce charging?
So we had proposed to put this directly onto solar panels is what we're intending to do in the phase 2 of this SBR activity. And so being able to put it along the edges where you put your electron conductors was where we were going to focus to start. So instead of having to make all the electrons that are charging on the solar panels travel to an emitter diode away from it, you could locally discharge, and reducing your challenges with, you know, robustness over time. You have local discharging capabilities on the solar panels themselves. Internally, I don't know how much charging happens. We hope to do a MISSY experiment in the near future to hopefully answer some of those questions.
Okay, getting into MISSY is, that's one of our long-standing payloads, so that's great. So thank you very much for bringing this, and your contact information is on the top of the slide. So if anybody has ideas on how to help them, go ahead and reach out to them and give them some ideas.
All right. Thank you so much.
Thank you, sir. So our next experience is with Sheng-wey. Jim is going to take over from here for a little while. Take it away, Jim.
Thank you, Alan. Let's go right into our next one here. So the title of this next poster, I get to say a great acronym again, is Microgravity Research for Versatile Investigations, or MARVIN, a modular system for processing experiments aboard the International Space Station. Our presenter is Scott Gilly, a senior engineer at TechMasters, Inc. Let's run the poster, please. Stand by, we'll have a backup in a sec.
The Microgravity Research for Versatile Investigations, or MARVIN, is an experiment processing system targeted for scientific and commercial investigations aboard the International Space Station. The MARVIN system accommodates interchangeable experiment modules, which offer heating, cooling, internal fluids and chemical management, power, data recording, telemetry, and video utilities. Sample processing from 0 to over 1000 degrees centigrade is supported as a specialty capability of the MARVIN system to study the joining and fabrication of materials in space. Melting and solidification are essential manufacturing processes, and procedures such as soldering, brazing, and welding for fabrication and repair will be crucial to long-duration or deep space missions. Unfortunately, these types of solidification processes are poorly understood in a microgravity environment. For instance, conventional soldering techniques in previous experiments aboard the ISS produced highly porous joints that are mechanically weaker and have degraded electrical and thermal properties compared to their Earth counterparts. Envisioned specialty areas for MARVIN users include coarsening and directional solidification studies to characterize microgravity effects on material properties for liquid melt, gradient freeze, and/or vapor growth methods of processing samples. For instance, precision translation stages will be available to directionally solidify samples using innovative heating modules. MARVIN provides tailorable translation speeds, by selecting from among multiple motor and gearing configurations. MARVIN further supports material properties measurement, such as diffusion rate determinations and multi-phase flow system studies, by tailoring the sample module hardware. Additional capabilities include investigations into fluid management and chemical reactions using mixing chambers, motorized syringes, custom glassware, rotational mechanisms, thermoelectric cooling and heating, precision resistance heater modules with controlled heat up and cool down ramp rates, with processing set points from 0 to over 1000 degrees C. MARVIN achieves this flexibility using a comprehensive control system with standard interfaces to the instrumented sample modules. This arrangement provides precision control of experiments contained in ground-prepared sample trays. The trays are similar in size to 2 stacked reams of copier paper and can be 3D printed for low-power experiments and for ground testing and development. A metal tray version will be offered to manage heat rejection and containment requirements of high temperature and/or high power trays on orbit. MARVIN automates data acquisition and control through redundant computing systems, reducing crew interactions and maximizing sample throughput. It provides a flexible form factor using interchangeable trays with adaptive sample interfaces. The tray interfaces include a robust selection of sensors, such as thermocouples and RTDs, analog and digital I/Os, USB, Ethernet, power and video functions. A wide range of solidification and heating experiments can be conducted using investigation-specific trays within the main housing or as external processing chambers using a breakout cable system. By providing a relatively large form factor and adaptive sample interfaces, a large variety of microgravity experiments can be conducted using investigation-tailored experiment trays. MARVIN has been designed to operate in the ISS maintenance work area, the Microgravity Science Glove Box, the Life Sciences Glove Box, or as an express locker. For more information, please contact Mala Thompson or Scott Gilly at 256-830-4000.
All right, excellent. Thank you. Thank you, Scott. So interesting project. Everyone out there listening, please don't hesitate to throw us some questions. And it looks like Scott's coming on screen there. So good to see you, Scott.
Hey, Jim.
So we don't have any questions for you right now. So why don't you take a little bit of time and talk a little bit further about any particular aspects of this that you like?
Okay, sure. We think the big attraction of this system is the interchangeable tray system. Modest size experiments could go up in these trays. We can also do custom modules that are a bit larger. For instance, we were the original developers of the PFMI and SUBSA hardware, which were larger in scope on their thermal processing chambers. Our particular specialty is high temperature processing. We've done several experiments up on the ISS as an implementation partner, both through Marshall Space Flight Center as well as recently Remus through Johnson. That can process up to 850 in the SUBSA. We've extended that capability somewhat for MARVIN, shifted over to a ceramic bore tube rather than the stainless tube for SUBSA. Gives us a little bit more reach on the temperature. The control system is modular, so that in this particular presentation, the MARVIN control module, MCM, is there on the right side of the upper left graphic of the 3D rendering up there within the MSG. And that is the focus of this particular paper, is the MSG as host. MSG has a nice selection of heat transfer, power capabilities, vacuum, nitrogen supply, and is operated for, I guess, close to 20 years now. So a lot of experience base with that particular host facility. One thing there towards the bottom of the presentation, you can see all of the different modules that will be supported for analog IOs, digital, actuators. You see a couple high precision motors down there in the lower left of the central portion, along with some linear actuators. We've done directional solidification studies with PFMI as well as Richmond furnaces back in the 90s. So we're experienced with both the hardware to process these high temperature experiments as well as the containment systems. All of this has to be safely contained. So a lot of that comes down to proper material selection, whether it's quartz, which you can do hermetic, torch sealing with to maintain integrity of the sample and avoid contamination of the atmosphere. Alumina, we've worked with sapphire. Recently, we're looking into polycrystalline alumina as a cheaper alternative to sapphire, not as transparent, but translucent. Also a big attraction of this is 3D printing capability, at least for the ground trays. These are of a size that some of the larger 3D printers can print these, and that is a lot of the photographs in this particular presentation are ground units that were 3D printed back in our lab. So upper right, you can see there's a very nice microscope system that can be utilized in the MSG. Navatar Resolve 4K can go down submicron on the feature resolution. Relatively high frame rate. It has a camera that you can select an area of interest to focus down in on and get the frame rate up to approximately 100 frames per second. So Tech Masters will be supporting these with our Telescience Center here at our Huntsville location. We have experience running samples from this, multiple samples. This has been selected for implementation already with our maiden experiment for this will be later next year. So the control system and the microscope will be up there for subsequent investigators to use as ancillary equipment provided by Marvin.
Excellent, thank you. I wish I could ask you an intelligent question, but I'm not there.
No problem.
It's very cool stuff, and you've been doing it a long time. So congratulations, and we don't have any other questions. So unless there's anything else,
I think that's it.
All right. Well, great. Thank you so much for being with us, Scott. We really appreciate it.
Thank you very much.
Great. So our next slot is a withdrawn poster. So we're going to take a little break. We will come back at exactly 3:50 Eastern time. Please come back. Thanks. All right, and we're back. So our 3:50 p.m. poster is Maturing Space-Based Precision Metrology with Atom Interferometer Studies Aboard the ISS. Our presenter for this is Jason Williams from the Jet Propulsion Laboratory. Let's run the poster, please.
Hello, and thank you for joining me to discuss atom interferometry, applications of this quantum sensor in space, and the recent first demonstration of AI on orbit in the Cold Atom Lab. I am Jason Williams, a member of the Quantum Sciences and Technology Group at the Jet Propulsion Lab. For Cal, I am the Flight System Lead and a PI for AI Studies in Space. Interferometry is a powerful tool for precision measurement. The shining example of this is the recent detection of gravity waves with the optical interferometer in LIGO. Here, laser light is split to propagate along two paths, and the interference measured after the beams are recombined give precise feedback of the environment that the two light beams encountered. Atom interferometry is highly analogous, only here it's the atomic wave functions that interfere, and laser pulses act as beam splitters and mirrors for the atoms. Shown in the upper left panel is data, actually images of the atoms from the Quantus Drop Tower experiment in Bremen, Germany. You can see the atoms separating, recombining, and the resultant interference pattern as the atoms free propagate thereafter. Capitalizing on cold atom technologies, which use laser and evaporative cooling techniques to reduce the temperatures of gases to microkelvin temperatures and below, atom interferometry is one of the most promising quantum technologies for space applications. In a nutshell, this can be understood because novel cooling techniques open opportunities for reaching unprecedented low temperatures in space. Low temperature gases allow freefall atoms to interact with laser for longer times, and in fact, the sensitivity of an AI to inertial forces scales quadratically with the time the atoms can interact with the laser in freefall. Applications of these ultra-high precision quantum sensors include geodesy and planetary science, navigation, and even fundamental physics such as gravity wave detection, sensitivity to distinct dark energy candidates, and a probe for the violation of fundamental tenets of modern physics. If you look at the lower left-hand panel of the poster, you can see the compact hardware of Cal, where the lasers, current drivers, electronics, science module, and control system are all housed in a so-called quad locker, which is about the size of a mini fridge on its side. The science module is a bit of a Russian nesting doll, starting with a cold quantum physics package in which the atoms are cooled. This package is housed in a water-cooled optomechanical platform which robustly holds the cameras and components to deliver light to the atoms. The optomechanical platform is then housed in two layers of magnetic shields to minimize the influence of the changing field as the ISS orbits the Earth. It's this science module that was recently successfully replaced on orbit to enable the new AI capabilities in Cal. The additional single locker houses power electronics but also holds the atom interferometer laser, which operates at 785 nanometers. And the Cal team that made this all possible is shown in the picture below. Cal was launched to the ISS on May 21, 2018, on board the Cygnus OA-9 rocket. After install by astronaut Ricky Arnold, and a three-month commissioning phase commenced. Results of commissioning included the first demonstration of BEC in orbit, demonstrated cooling of a quantum gas to sub-nanokelvin temperatures, and detection of an ultracold gas after 1 second of free expansion. As shown in the center panel, these results were published in Nature this summer. Please see the presentation by David Aveline on September 22nd, detailing these results. After commissioning, Cal demonstrated itself as a truly multi-user facility by supporting science for 7 flight PIs, 3 of which are Nobel laureates and founders in our field. In addition to commissioning results, Cal has demonstrated cooling of dual-species rubidium and potassium mixtures, delta-kick cooling to picokelvin temperature regimes, demonstration of the first bubble BEC, and demonstration of atom interferometry for the first time in orbit. AI in Cal was enabled by an on-orbit replacement, as we said, of the science module that was launched to the ISS on board the SpaceX Falcon 9 rocket on December 5, 2019. This complicated upgrade was successfully executed by astronauts Christina Koch and Jessica Meir, and shortly thereafter, BEC was reestablished in Cal. The AI is implemented as shown in the upper right quad of this panel, where the AI laser passes through a window on the atom chip to enter the science cell. It intersects with the atoms in freefall and executes a 3-pulse AI scheme as shown to the right. Images of the atoms are used to determine the relative phase of the AI by measuring which output the atoms occupy. This scheme was successfully demonstrated in late May of this year, where by scanning the phase of the final laser pulse, it was possible to vary the population of atoms in each AI output. Hence, the data you see here is the first demonstration of matter wave interferometry with ultracold atoms in space. Going forward, Cal aims to achieve dual species differential atom interferometry measurements and a number of both applied and fundamental physics experiments based on this one-of-a-kind quantum sensor. Thank you.
All right. Again, very in-depth, interesting stuff. And we've been talking a lot about Cal during this conference. It's very cool. So we have with us Jason Williams here from JPL. Jason, welcome. Good to have you here. So again, right now, unfortunately, we don't have any questions and I don't really have anything for you. So if you have any additional comments or things you'd like to talk about with this, please feel free for the next 4 minutes or so. And if something comes up, I'll interrupt and ask it.
Okay. Yeah, thank you, Jim. So with the time allowed, first, I would like to give thanks to our sponsors that have supported us. The ISS Program Office and also the Biological and Physical Sciences Division of NASA's Science Mission Directorate have been incredibly helpful, both assisting and in many ways helping guide us through developing this unique Cal system. As you said, there's a lot to this poster. There's been a lot of talks on the Cold Atom Lab. It's been operating for over 2 years in space now, been producing ultra-cold gases. What we mean by that is these are quantum gases, where in many ways the wave-like nature of the atoms becomes either relevant or sort of dominates the physics that we're exploring. And atom interferometry is a very strong example of quantum effects in atoms and how those can be used for future technologies. I really love this technology. It's one of the unique aspects of quantum physics that I found very intriguing when I was first learning about it in grad school. But this is a truly single particle phenomena. So you can send one particle through the interferometer and the atom actually itself splits to travel along two paths and interferes with itself. So we're using matter waves and the interference of matter waves for precision measurement. In the time remaining, I also want to point out, since many of the presentations are available, as you mentioned, there are a number of other related presentations by our colleagues at JPL and also the various other PIs on Cal. So if you get a chance, please check out the plenary talk about the Cold Atom Lab by Kamal Ojuri, who is our project manager, and Rob Thompson, our project scientist. Also, during the first session yesterday morning, chemistry and particle physics research capabilities. There was a talk by David Aveline, as I mentioned, that reviewed the commissioning results that we got right after Cal had launched. And those results also you can find, they were published in Nature in June of this year. And that Nature cover you see there is related to this research. So that was good. Mateo Brassica is also in that session talking about blue box potentials. This is a new, very relevant technology for space, where you can actually trap atoms inside a box instead of a trap. And if you do that on ground, the atoms will just drop to the bottom of the box. But in space, you'd have a truly confined system, but the atoms are free in the center of the box. So that's interesting. Also talked by Makan Heg about some space quantum optic experiments, going to the other side of quantum technologies, where we're looking at quantum information, quantum key distribution, things like that. So I see that I'm running out of time, but yes, please check out all the related Cal posters and presentations.
Excellent. Excellent stuff, Jason. Thank you so much for being here. Thanks for all that. Yeah, and again, Cal has just been all over this event, and it's really good to see such a productive piece on the station. All right, with that, I'll turn it over to Alan for our next segment.
All right, thank you, Jim. Our next presentation is on droplet microarray, a miniaturized platform able to confine liquids in low gravity conditions for various biological experiments. Anna Popova from the Karlsruhe, I hope I said that right. She can correct me, she will. The Karlsruhe Institute of Technology will be our presenter. So please play the slide.
And I'm happy to present you my poster titled Droplet Microarray, a miniaturized platform able to confine liquids in low gravity conditions for various biological experiments. So I will start with a small introduction. Performing biological experiments in space is very important in order to understand how space conditions like gravity or space radiations is influencing our physiology. In addition, diagnostics and point-of-care applications are important in order to enable fast hands-on tests of fluids and cells from individuals. Any biological experiment or test is taking place in liquid environment. However, it's a challenge to confine liquids in the conditions of low gravity. In commonly used petri dishes or multi-well plates, liquid is confined due to the gravity. So in our laboratory, we have developed a droplet microarray platform, which is based on hydrophilic superhydrophobic patterning. It is a planar transparent array, if you can see, as you can see here on the left, which contains hydrophilic spots located on superhydrophobic background. So due to the extreme difference in wettability of hydrophilic and superhydrophobic surfaces, the array of separated homogeneous and stable droplets is formed spontaneously on this surface. The size and shape of the spots as well as design of the pattern can be easily adjusted, and the array of droplets can be created by using low volume dispensers. However, it's not necessary to use any specialized equipment, and due to the effect of spontaneous droplet formation, the array can be formed spontaneously simply by applying of a solution or suspension of cells on the pattern surface. So due to the absence of the physical borders, we can reach unlimited miniaturization and go down to nanoliter volumes without droplets. These droplets can be used as miniaturized wells for culturing cells. We have tested our platform with different types of cell culture models, with adherent suspension cell lines, with primary cells. Also, we can use it with 3D cell culture, using it for spheroids or scaffolds like hydrogels, as well as stem cells and other types of cells. On the right side, you can see how we use it for high-throughput screenings. We usually preprint compound libraries on the chip, and afterwards we can store those libraries and dispense the cells on the chips containing the libraries, performing the screening on the chip. We have demonstrated the feasibility of using our arrays for high-throughput screenings, as you can see on the right bottom, using the commonly used HeLa cell lines as well as a patient-derived primary cells. We also used our system for confining zebrafish embryos in 5 microliter droplets. So taken together, the droplet microarray is a versatile platform which can be used for various high-throughput biological applications in miniaturized format. And the main feature which makes the droplet microarray platform essential, suitable for performing biological experiments in space, is its ability to confine liquids in low gravity conditions. The array of the droplets is stable, independent of position of the slide. Slide can be turned upside down and rotated, and the droplets are not affected by that. So we believe that droplet microarray platform can be applied for multiple applications in space, from scientific projects, screenings for agents protecting from the harmful effect of space radiation, for example, or to diagnostic tests like testing individuals' fluids containing bacterial infections for sensitivity to given antibiotics, for example. We have not conducted any experiments in the conditions of low gravity yet, but we're very eager to do so, and we would like to raise awareness and interest of scientists, engineers, astronauts, and people who have experience working on in the space and we wish to create collaborations, get inspired, and inspire other people with a final goal to apply our technology in space for making an impact, for creating tools and achieving the goals that are not possible to achieve at the moment with other platforms and technologies. Thank you very much.
All right, Anna, thank you. If you could activate your camera and your microphone, we'll move on.
Hi.
There she is. All right. So right now we don't have any questions. So is there something that you would like to expand upon that you did not get to talk enough about in your presentation on the poster? If not, I have some dummy type questions I can throw you, softballs.
Yeah, sure. Just go on with that.
Only want softballs. Okay. The first one is that, you know, the hydrophilic versus hydrophobics and creating a mesh or a matrix on a sheet it seems rather obvious, but it's not. Your guys are the first to come up with it that I know of. Why is it not so obvious?
Well, that's obvious to wanted to have something like this, but it's not very easy from chemical and topographical perspective, because in order to have such a difference in hydrophilicity and hydrophobicity, you have to have combination of certain porosity, roughness of the surface plus chemical modifications. So there is actually a lot of research going on that, and there is a lot of surfaces, but not all of them are that stable. So not all of them allow to form such droplets simply by dipping in and have essentially a stable droplets that are not merging even by shaking. Also, we perform biological screenings on them, so they have to be biocompatible. They have to be transparent, which is very difficult for porous and rough surface to have these properties, and transparent, of course, for microscopy-based analysis. So combination of these features are actually quite difficult to find. So, and there is a few years of research been into these platforms, and for the past almost 10 years, there's a lot was changed in chemistry and coatings that we have been developing.
All right. Would you expand a little bit upon the non-space utilization of this? As we miniaturize our ability to do sample analysis and need less and less and less sample to perform analysis, what is the utilization in this in a ground-based type system?
So, I mean, the obvious thing that we're focusing now on cell screenings is ultra high throughput screenings that's performed for drug development, especially the primary screenings that performed with hundreds of thousands of compounds. And of course you need, yeah, that's a huge experiment. So as lower you go, as more experiments you can conduct. Those are also horribly expensive. Also now people go to more physiologically relevant cells, so they're not available in such amounts. So we see this kind of chip technology is being well welcomed when it's developed to its maturity in pharma companies, and we already got a lot of interest in that area. Another thing we're seeing for is for personalized screening. So the results are presented in the lower right corner is a testing of actually patient-derived cells, where you can test, for example, biopsy cancer cells to see in experimental way in like 2-3 days to which drugs or drug combination they actually respond. And we have an ability to test very small amounts of patient material, also with the large libraries with of compounds and anti-cancer drugs. So that's another applications. And what I also wanted to add for the space applications, there has been a lot of research, of course, on tissues, on cells, on in vitro systems. So our system is in vitro system. It's pretty flexible in terms of which in vitro model you use. So we can use different cell models in 2D and 3D. We can grow spheroids, we can use scaffolds, so it's versatile. On top of it, we use also zebrafish embryos, as I mentioned shortly, in 5 microliter droplets. We never use like C. elegans or other animals, but it's absolutely possible also incorporate them in the droplets. So in comparison to what I've heard and read, what using in space, our platform can allow high throughput screenings. So not testing a few conditions, but actually testing, for example, the drugs which can protect from radiation. And in high throughput manner, also on a physiologically relevant tissues, for example. So we see our USP in the area of screening more than just to see the effect of some space conditions.
Well, thank you very much. This is really interesting. I'm glad you guys are doing it and moving forward. And it began to sound a little bit like tissue structuring when you started talking about 3D and using that. So thank you very much. And we really appreciate your presentation. Time to move on to our next presentation. And this is water conservation on the ISS, how the amount of water provided to plants can be reduced. And this was brought to us by Christine Brilhart of Midland Public Schools. Let's play her slide.
Thank you for choosing to learn more about our project. Our presentation is titled Water Conservation on the ISS, how the amount of water provided to plants can be reduced. Now Jessica will share some background information.
Hello everyone. Our Growing Beyond Earth project with Fairchild and NASA aims to help find plants that can survive with reduced water consumption. This is because NASA astronauts on the ISS need to conserve water, as it is extremely expensive to carry water there, and wastewater from fuel cells and personal hygiene, and they have a significantly reduced water consumption rate. Without these measures, the ISS would need 40,000 pounds of water delivered yearly for just a minimum of 4 crew members. So in order to continue this water conservation further, save money, and make water available for other activities on the ISS, it is important to find suitable plans for water conservation. Next, Margaret will present the summary of our experiment.
Hi everyone. The goal of our experiment is to find out which lettuce cultivars is the most suitable plant that can cope with less water and provide the most biomass overall compared to ideal conditions. 4 lettuce cultivars were tested in 2 28-day trials, including red romaine lettuce, butterhead lettuce, lettuce Sulcindus, and butter crunch lettuce. Our water conservation principle is to use 30% decrease in water consumption for the experimental cultivars. Each week, we totaled the amount of water used for the control group, multiplied that amount by 0.3 to determine how much water to reduce. We hypothesized that at least one lettuce cultivar could cope with 30% less water and provide at least the same amount of total edible biomass as the control counterpart. Next, Temi will present methods and materials.
Hello everyone, I'm going to be discussing the methods and materials we used to collect the data for our experiment. Data concerning the growth of the plants was collected daily. Many variables were measured and recorded, such as plant height, plant width, plant depth, number of leaves, and plant health. The amount of biomass recorded only includes the leaves trimmed to the stem of the lettuce cultivars, which was considered the edible biomass that an astronaut would consume. Thanks to Fairchild, all of the materials needed for data collection were already provided. We also used a digital moisture meter. During the control trial, water was added only when the plant tray felt dry, whereas during the experimental trial, water was added only when the digital moisture meter indicated a low moisture level. Next, Saba will discuss the results of our experiment.
Good day, everyone. The results of our experiment show that with a 30.7% decrease in water consumption, there was a 72% decrease in the total edible biomass and a 26% decrease in the total number of harvested leaves. Our graph 1 indicates that the edible biomass of the red romaine is decreased by 75%, butterhead by 70%, alkindness by 85%, and butter crunch by 57%. Our graph 2 shows that the total number of harvested red romaine leaves is decreased by 43%, butterhead by 34%, alkindness by 25%, and butter crunch by 0%. These results imply that the water conservation has the least impact on the butter crunch lettuce than other varieties. In the same token, butterhead is ranked number 2, alkindness is number 3, and red romaine is number 4. Our graph 3 reflects an overall picture of the water consumption in the experimental group and the moisture readings of the plant tray. The water conservation method used in the experimental group was followed, but with some exceptions. This resulted in a 0.7% increase in the predeterminate water reduction. Next, Margaret will wrap up the presentation.
In conclusion, while none of the experimental cultivars produced at least the same amount of edible biomass as their control counterparts, Buttercrunch lettuce stands out as the most resilient cultivar, as it is the best at coping with less water. It produced the most edible biomass and has consistent growth in terms of the number of leaves and plant dimensions. Plants use water for photosynthesis, but it depends on the amount of water given. Overwatering crops in the ISS space garden can cause fungus to grow on plants and affect the ISS crew members' health. Underwatering may change the rate of photosynthesis, delay fertilizer and growth media to dissolve, and result in slower plant growth. Therefore, the results of this experiment provides a step further on water conservation, on crop cultivation, and thus aid scientists at NASA Kennedy Space Center to find suitable crops to add fresh food to astronauts' diets. Thank you for your time. Any questions?
All right, Christine, if you can activate your camera and your microphone, we'll go ahead and jump into this. There you are. Howdy. Hi. Distancing and mask. All right. So now, as we wait for a couple of questions to come in, I don't like lettuce. So why is this important to me?
Well, lettuce or fresh food provides vitamins that dry foods or produced products don't. Like, for example, vitamin K is not a reproducible vitamin, so plants or lettuce will be able to provide astronauts with that food.
Whether they like it or not.
Yes.
You will eat your lettuce, kind of like Popeye and spinach. Okay, I was going to ask you a question about one of these folks, but I saw them as a project sponsor. It looks like you're doing some work with the veggie guys, either at Kennedy or in Orbitech. What is your activity with the veggie guys?
I think Jessica can answer that question.
So we actually worked with them so they sponsored us and gave us all of our materials and everything, and we actually did the presentation. We did the same presentation previously for that. So then they invited us to come do this.
Okay, they're great guys. They're great program. You learn a lot from each other. Another very, very important question, and I'm going to expand on it a little bit. So you reduce the amount of water provided to the lettuce. Number one, what does that do to the taste of the lettuce? And number two, which of the lettuces taste best?
So I actually wasn't there when we harvested the lettuce, but Margaret can talk about that.
Right. So from our observations, there wasn't really a difference in the taste of the leaves, but I think the best tasting cultivar was the butter crunch lettuce, which I believe is the best plant and the most resilient plant from our experiment.
All right. So why lettuce? Why did you guys go off and use lettuce instead of something else?
Well, for the Fairchild project, when we decided to go into this, they had given us the lettuce, so we didn't decide on the type of lettuce. They were the ones that gave us the lettuce to try, and the seeds.
Mother of invention. You had some lettuce, and you had to do something with it. Nobody else would eat it, so you decided to play with it. Okay, so this is almost the reverse of hydroponics. Thinking about it a little bit. So have you gone in and done any studies of what was done to plants to help them to grow better in an arid environment, like in a desert or a semi-desert environment? And how does that correlate to any of the work that you've done and what you've learned about reducing water supplies?
Well, I think our project was based more on overwatering because in the previous time we did this, which was last year, we ended up overwatering the plants. So they grew green algae and mold, which wasn't so good. So this year we decided that we would try to figure out what would happen if we reduce the water amount and slightly underwater the plants.
Get both ends of the spectrum. That's a good way to do. Establish your boundary conditions is the way they describe it in some places. So now, are you guys talking with our veggie friends or anybody else about the difference in your results if you were in microgravity?
I believe not.
All right. Well, very fascinating, and I'm glad we were able to bring you guys in. And I have a minute here to spare. So what I really wanted to do is to explain to our audience that we don't go off and put our students into a little class by themselves and let them talk just to themselves. We mainline our student researchers as much as possible so that the senior folks can understand what the younger people are doing and maybe even learn from it. One more question just while we're at it. Did you evaluate evapotranspiration? Do you think the lettuce cultivated with less water will be stronger to face high energy rays?
Well, based off our observations, it seemed like with less water, the cultivars are a lot weaker, and the plant dimensions and the biomass are a lot smaller, actually 3 times less than the control group. So, although we haven't tested it based off our observations, probably not.
All right. Well, thank you very much. This has been really interesting, at least to me. And I know a few other people got really interested in it. So thank you very much. And now it's time for our next presentation, which happens to be a blank spot in the agenda. So we're going to come in and let you guys have a little bit of time to take a potty break, have a drink of water, cup of tea. It's your afternoon cup of time. And we'll come back at 4:30 with the next presentation, and Jim will be emceeing that.
Okay, everybody, it is now 4:30 and time for our next poster. So let's get right into it. This one is called Leveraging Microgravity to Screen Oncoselective Messenger RNAs for Cancer Immunotherapy. Our presenter is Yusuf Erkul, co-founder, president, and CSO of Kernel Biologics, Inc. Let's run that poster.
My name is Yusuf Erkul. I'm a medical doctor by training, and I have research background in immuno-oncology from Merck. I'm a co-founder of Kernel Biologics, which is developing oncoselective messenger RNAs for cancer immunotherapy. At Kernel, we've been working with cancer cell ribosomes and translational landscapes of cancer cells with the goal of developing oncoselective or cancer cell-specific mRNA therapies. Messenger RNA itself has recently garnered a lot of attention from both media and the general public because it allows us to actually safely and effectively express proteins of interest for therapeutic purposes. This is a novel modality that generated a lot of excitement because it's quite effective in driving phenotypic changes in vivo. And at Kernel, we are developing the next generation of messenger RNAs by making the messenger RNA specific to cancer cells. With this ISS National Labs and Boeing supported study, we are aiming to actually bring some of our oncoselective messenger RNAs to the space station with the goal of benefiting from taking advantage of the microgravity environment in the International Space Station, because microgravity has previously been shown to affect some key mediators of ribosome biogenesis. And these key changes are closely related to some of the key changes that we see in cancer cells. For example, cancer cells, when they lose p53, typically also have key changes in their ribosomes, and they have significantly altered translational landscapes. This is something that the Kernel platform is based on. And microgravity really offers us with a great opportunity to screen some of these messenger RNAs to de-risk them before clinical development. We have, within this research study, switched to the BioServe BioCell plate system, and we had to do a number of optimizations for the experiments that will be launched next in about 2 weeks from now with NG14 mission. These changes involved switching to a flight-ready cell culture system. BioServe plate provided us with these 12 well plates. We typically use Petri dishes with 12 wells in them that allow us to actually test multiple messenger RNAs and multiple replicates of the same messenger RNA simultaneously. However, with this space-ready system, we realized that we had to use fully concealed and fully enclosed systems that have initially posed some challenges because as we did, for example, injections of cells, there was some shear force that was affecting their viability. Then we improved the needle gauge size and the speed of injections. We then had to face the fact that in order for us to seed these cells here on Earth and then ship them to the space station, they have to endure in cell culture for up to a week. And that is why we have to optimize the culture conditions, especially for the cancer cells. We have noted that if we seed too few cells, they don't grow at all. If we, for example, seed too many cells, they get overly confluent quickly before our experimental timeline. So we had to play around with culture conditions, with temperature of these cells, and we had to also improve their growth media to actually support their continued growth during the course of the planned study. And after doing all those changes and after also optimizing how we do the mixing of the reagents in these special plate systems, we have now established our final protocols, and we are currently ready for a launch that is coming up in 2 weeks. We had also had to deal with the plate reader issue. In the International Space Station, there is a plate reader that allows us to do luciferase measurements. However, this plate system is not as sensitive as the typical plate system that we use here on Earth. Therefore, we had to switch to a different system. We had to improve our delivery agents to really boost the signal and make sure that we have sufficient signal to noise ratio that would allow us to actually perform the experiments in microgravity. So having completed all those key changes, we are happy to note that we are in a position to now actually launch this payload. If you have any questions about microgravity, ribosomes, or our oncoselective mRNAs, I'd be more than happy to talk to you. Please don't hesitate to reach out to me at yusuf@kernelbio.com. That's my first name, YUSUF, at kernelbio.com. Thank you so much. Bye-bye.
Excellent. Thank you, Yusuf. Very interesting work, and I'm sure you're looking forward to actually getting up into space. So please do, if you haven't already, bring yourself up on screen. I see you coming up. There you are. Good to see you. Excellent. So we are waiting to see if there's any questions for you. So in the meantime, please feel free to talk a little bit more about your project and anything else you got going on.
Absolutely. I can provide some background information about the source of this specific study and why we're targeting the microgravity setting for our messenger RNA testing. Previous studies on cancer cell ribosomes have shown significant changes in both ribosomal RNA chemistry and ribosomal protein content that helps us understand why the cancer cells typically end up overexpressing certain oncogenes and silencing certain tumor suppressor genes at the mRNA level. In addition, these changes actually resemble some of the changes that we see under microgravity that are reported by space biology groups that were doing studies, for example, under simulated microgravity. We see key players like fibrillary emerge on both ends. Both the microgravity research and cancer biology research indicate key mediators of ribosome biogenesis as being altered under malignancy based on changes in p53 expression, but also under microgravity based on more general changes that are induced when human cells or eukaryotic cells are brought to the space or any simulated environment that resembles the microgravity in space. So at Kernel, by using this, this scientific understanding of ribosomal changes under microgravity, we wanted to bring our cancer cell-specific messenger RNA platform to the International Space Station to allow us to actually do further testing, in a way stress testing, to ensure that our oncoselective messenger RNAs are indeed oncoselective, despite some unusual changes that can be seen, for example, under microgravity. I have previously spent many years in oncology field, some of which was spent at Merck, working on preclinical models for oncology drugs. I've been involved with drugs like pembrolizumab for the preclinical models. I've also been involved with PARP inhibitors. In both cases, what we're trying to achieve in preclinical space before we bring assets to the clinic is, can we de-risk them? Can we identify targets that are better poised for further investments than the ones perhaps that we don't want to advance to the clinic, because clinical development is very expensive. It's also costly on the human side because patients typically have high expectations for experimental treatments because this is, in many cases, unfortunately, their last chance. So we want to make sure that we do our best in de-risking and improving our selectivity profile of our messenger RNAs before we start our first in human phase 1 studies. And this study was really geared towards making sure that we can identify conditions that are known to alter ribosomes in normal cells and in cancer cells, and to do further testing to ensure that we identify very promising lead assets for further development.
All right, excellent stuff. Thank you so much, Yusuf. We really appreciate your being here. And again, if anybody wants to look at his poster in more detail, just go on to the ISSconference.org site click through and you'll be able to find it. So thank you very much again for your time. Really,
Thank you for
really
having
great
me.
having you here.
Great pleasure.
Thank you. Okay, so unfortunately, our next poster had to withdraw from the conference. And so we'll be taking a 10-minute break, but we'll come back at 4:50 Eastern. See you in about 10 minutes. Thanks, everybody. Hi, everybody. Just wanted to let you know that we actually are going to run the poster that was originally scheduled for 4:40. Although the presenter could not be here, we're still going to run that poster. So listen to this one, and then we'll come right back for our next poster originally scheduled at 4:50. So let's go ahead and run the transcranial Doppler poster. Thanks.
Introduction. Parabolic flights in patients with invasive Ommaya reservoir drains show increase in ICP in periods of relative microgravity. Long-duration spaceflight, such as the ISS and the syndrome of spaceflight-associated neuroocular syndrome, raise the question of the effect of global ICP and other factors on this syndrome. TCD, or transcranial Doppler, is a non-invasive way to assess cerebral circulation, as well as inferences about intracranial compliance, elastance, relationship on Earth and ISS. We searched the NASA and the National Library of Medicine databases, as well as a neurosurgical population at risk for hydrocephalus requiring invasive ICP monitoring. Regarding these databases, we could only find one head-down study on Earth studying TCD on intracranial pressure. We subsequently reviewed a series of aneurysmal subarachnoid hemorrhage patients with hydrocephalus with EVDs and TCD resistance and pulsatility indexes, including RI and PI. These values that approach 2.0 and 0.8 respectively were defined as altered ICP compliance versus elastance relationship and visualized on the TCD waveform. Normal TCD waveform was defined as a normal waveform with 3 peaks, an abnormal waveform of poor compliance showing triangular waveform. From 2008 to 2013, 5 patients actually developed delayed hydrocephalus and had elevated RI and PI values at any point during their initial hospital stay. Here we have a figure of one of those TCDs with these indexes. TCD may serve as a potential low complexity and accessible method to assess and monitor ICP compliance, elastance relationship during SANS and ICP development in conjunction with other multimodal monitoring methods in microgravity, such as optic nerve sheath diameter, globe shape, optic dysedema, and optical coherence tomography for both microgravity travelers and patients on Earth.
All right, that was a poster from Kelly Gassey at the Mayo Clinic in Florida. So now we're going to run, it's just about 4:50. So we're going to run our next poster, and this one is called the Space Frontier Studio Kibo, the face-to-face interactive live communication system and live streaming experimental program. Let's run that poster.
A digital creative company based in Tokyo launched the Space Frontier Studio Kibo in August 2020 as one of the projects of the Joint Research and Development Program JSPARC with Japan Aerospace Exploration Agency. This space media business project aims to open the door to space where it is still closed and limited to all people by the power of technology and design. The laptop terminal installed by the round window facing the Earth of the Japanese experiment module Kibo on the International Space Station is the main equipment of the newly constructed the Space Frontier Studio Kibo. Messages uplinked by the Grand TV studio located in Tokyo are shown on the onboard screen. Videos showing the interior of the module Kibo are downlinked to the Grand Studio. This is the world's first face-to-face participatory two-way live communication system linking space with Earth. This system is utilized to produce and deliver TV broadcast programs. The program was arranged and progressed with the 90 minutes ISS orbit around the Earth. As the ISS travels in daytime zone, various communication experiments are conducted linking between the module Kibo and the ground studio. During the nighttime zone, the Perseid meteor shower was also filmed and broadcast live by the ground studio. Let's get in closer to each challenge. Challenge 1. Development of face-to-face two-way live communication system outside of the existing intranet station. The ISS connects on an intranet with the Tsukuba Space Center, and it is not possible to import external data into the intranet. Our system is based on the mechanism of converting external data into two-dimensional codes, capturing code, and then reconverting into data to transfer real-time external data. It enables external data to be imported on the real-time basis while maintaining secure of the intra-network. By installing the application on the laptop terminal in the module Kibo, the data emitted by the ground studio in Tokyo are received through the system booth in the Tsukuba Space Center by the application running on the laptop terminal. It can be operated on the real-time biases from the ground studio in Tokyo in arranged with the progress of the TV program. Challenge 2. Fabrication and operation of variable ND filter for optimal filming of intra and external vehicular objects with different illumination levels. Due to the ever-changing intensity of sunlight, we have to adjust different illumination levels for the screen on the onboard laptop terminal and the Earth as viewed through the round window of the module Kibo with an optimal exposure required real-time regulation. We therefore fabricated a variable ND filter with double polarized film dedicated to filming the video through the round window of the module Kibo and transported it to the ISS. The user need only roll the closer filter to one side in other to smoothly reduce the light. In addition, the position and the filming angle of the camera and laptop terminal are verified with the 3D simulator. Thus, the camera was precisely installed in short time. Challenge 3. Broadcast programs in arranged with ISS orbit, daytime, nighttime zone, and acquisition of signal, loss of signal. We visualize the data of time zone of ISS, sunrise and sunset times, acquisition of signal, and loss of signal zones on a single world map. These visualized data are used for producing broadcast programs. The internal and external vehicular camera angles and the relative orientation of the sun were simulated by using our original 3D simulator. Both the onboard crew and the viewers on the ground could thus observe the rising sun together during the broadcast. As a future vision, we plan to update technical challenges, such as shortening the delay time between the ground and the ISS, improving video quality, and supporting AR/VR technology. At the same time, we will take a challenge of creating content that everyone in the world can sympathize with, and we will provide opportunities for more people to express their thought in space. And in order to continue this project for a long time, we plan to build a business model as a commercialization. Thank you.
Okay, very interesting. I'd like to bring up Rumiko Nango. Hello, welcome. Good to have you here. Please unmute. There you are.
Thank you.
Excellent.
Hi.
Anybody watching, if you have any questions about the Space Frontier Studio, please submit them. Until we have those questions, if one comes up, Rumiko, I will interrupt you and ask. But until then, please feel free to talk a little bit more about the project. Well, actually, wait. Two questions just came up. Is there a recording of the live event from August 12th available online?
Yes, our movie will be archived on the official website. And right now, like a little by little, like a part by part, we already on show on our official website. but it
Okay.
will be open on YouTube. You can see you can search through the YouTube, and you can see some part of the program as well.
Excellent, excellent. Okay, and we do have another question. So how does this communication system differ from the sort of real-time communication that NASA uses between ISS and Earth now?
Good question. But what we are trying to do for this time is we want to access outside of the intranet between the ISS, NASA, and JAXA, because we have outside of studio in Tokyo, which is like far from the JAXA as well. So what we are trying to do is we put some input from the Tokyo studio through the JAXA and through NASA and go to the ISS. And the intranet is prohibited to access outside of the, the access from the outside of the network. So that is one of the challenges. So which is, it's not the new point is we can access from the studio in Tokyo to directly to ISS, which is through from JAXA and NASA.
Excellent, excellent.
Yeah.
Thank you. I think that answers the question. So we don't have anything else right now. So you have 3 minutes to talk more about the project.
So we're planning to do the next TV program show maybe within this year. So it's going to be all maybe in Japanese right now, but we're trying to do work for the for people can see more information or more interesting program for all the people. Yeah, so we are doing the second try within this year.
Okay. I really don't have much else for you. Was there anything else you wanted to talk about?
Actually, someone put online that it's in English side is not updated yet, so it has to be changed to Japanese /JP. I will put the link to the QA on the answer.
Okay. Yeah, you can do that when we're finished. What about educational outreach? Have you done any work with schools or universities on this project?
Not yet, but the first try is we're collecting a lot of videos from the regular people. For the kids and children and also babies and so on, so on. So what our purpose is bring those people to the space. So we show a lot of not astronaut people on the screen in ISS at the Kibo. So that is one challenge, but we are also trying to do more open for the public and open for the kids and schools as well.
Excellent. Okay. Well, thank you very, very much for your time, and thank you for coming on. It was an excellent presentation and a fascinating project. So thank you very much.
Thank you very much.
All right. And with that, I will turn it over to you, Alan. I think you're muted, Alan.
Well, you can see me at least.
I can.
Okay, so the next presentation is the Rhodium Scientific's DoD prototype process, enabling space-based R&D with the speed and quality required for commercial markets. The presenter is Olivia Holhaus, the founder and CEO of Rhodium Scientific. Now, we don't find her in our list of attendees at this point, so right now we're just going to play the video, and then I'll come back to you and tell you if you have a 5-minute break or not. So let's go ahead and play the video.
Hi, my name is Olivia Holzhaus, and I'm the founder and CEO of Rhodium Scientific. Rhodium is a biotechnology company providing science and mission support services for researchers across industry, academia, and government. Our role in the commercial space industry is focused on developing, implementing, and facilitating applied and discovery-based science in space with the quality assurance standards the industry recognizes. We believe that discoveries made in space are dependent upon upholding the same scientific rigor as is practiced in the client's terrestrial lab. As a current implementation partner to the International Space Station U.S. National Laboratory, our quality science first philosophy serves investigators and investors by providing unprecedented levels of quality assurance, expedited access to space, and cost-effective solutions for payloads across a wide range of scientific fields. Quality science first enhances the experimental reliability and enables product prototyping. It also supports the testing and manufacturing of products in regulated industries. This approach has proven successful as our company has completed multiple ISS missions this year, with several more slated for September of 2020 on NG-14. A majority of these projects have been contracted by the Defense Threat Reduction Agency, a combat support agency within the Department of Defense, also known as the DoD. The overall goal is to develop a microgravity research prototype process that integrates industry standards into the NASA Mission Integration and Operation process. This process actively attracts a wide range of biotech-based clients desiring to prototype their own microgravity-based products at the speed of business. In order to do this, Rhodium combines PhD-level expertise with NASA flight operation and safety expertise to develop projects that are both scientifically credible and flight feasible. This unique combination of in-house capabilities has allowed Rhodium to develop the quality, industry-compatible space process, a high-fidelity science process known as the Quick Space Process. The Quick Space Process was vetted by and is currently in use by the U.S. Department of Defense, Los Alamos National Laboratory, and multiple Tier 1 research universities. Overall, the Quick Space Process develops scientific projects at the speed of business with the appropriate levels of quality assurance necessary for commercial products of regulated industries. This process is being used in 3 of our current 2020 investigations. The first investigation is named Phage Evolution. Phage Evolution is a collaboration with Dr. Vatsan Raman at the University of Wisconsin-Madison and focuses on bacteriophages. Bacteriophages, or phages, is a virus that only attacks its bacterial host. The study explores the evolutionary rates of phages and their host range for targeting selected bacteria in microgravity. As microbial resistance to antibiotics is on the rise, so is the need for new therapies. This project can potentially develop new phage therapies targeting microbial pathogens. As such, the DoD desires a level of quality assurance from start to finish to ensure the results of the project are accurate and reproducible. The second process utilizing the Quick Space Process is named Rhodium Space Microbiome. This study is a collaboration with Dr. Armin Dicosa at Los Alamos National Lab. This project flew representative gut microbiome samples and exposed them to microgravity aboard the ISS. This was to determine how and what gut microbial changes occur during the 1G to 0G transition. As environmental shifts have been shown to alter gut microbiome composition, which can cause disease, this is of important interest to the DoD. The DoD is interested in scientific data that could support the creation of a diagnostic medical device, thus requiring the project to encompass full quality assurance protocols throughout the project lifecycle. The third project using the Quick Space Process is Rhodium in-flight biomanufacturing. This study is a collaboration with Dr. Mark Blenner at Clemson University and explores how various bioengineered strains of bacteria and yeast produce mission-critical organic products of interest. Full quality assurance protocols are being applied to this project as these products of interest solve point-of-need manufacturing issues regarding traditional supply chains in areas of remote operation. The key to our business success is to operate both effectively and efficiently when conducting research in space. Rhodium has a suite of flight-certified hardware called Rhodium Science Chambers. The Rhodium Science Chamber can support the needs of a biotech market. They can be configured for passive experimentation or for experimentation requiring gas exchange. If a client's experimental needs exceeds our current hardware capacity, we can quickly adapt by 1, certifying new hardware, or 2, working with other implementation partners as a hardware broker. Roading can provide a distinct time-to-flight advantage that positively impacts our client's ability to work at the speed of business. We look forward to working with you to help you move your research and development efforts to space with the quality you expect and use on Earth. Thank you for listening to my presentation. My contact information is at the bottom of the poster, along with our social media listings and hashtags. Thank you.
All right. So we have no panelists to answer any questions, and I'm not going to pretend that I can answer questions. So we're going to take a 4-minute break, and we'll come back at 10 after the hour for the last presentation. We've verified the presentation is there and the presenter is there. So take a 3-minute water break, and we'll be back at 5:10 Eastern Daylight Time, New York time. All right, so we're here for the last presentation of the day. It's been 5 hours and 20 minutes since we started this. It's been an interesting experience for all of us. Atif, thank you very much for being patient and hanging into this late for us. Atif Kusari is from Maxar Technologies, and his presentation is Demonstration of CubeSat Capture by a Novel Robotic System. So let's play the slide, please.
Hello, my name is Atif Qureshi. I'm with Maxar Technologies, and I'm here to present our proposed demonstration of CubeSat capture by a novel robotic system. I'd like to begin with a little discussion of the motivation behind this development. We see two key areas of application for this technology. The first being the need for space domain awareness in the cislunar sphere, which is being driven by NASA's return to the Moon. This is an area where we expect small distributed assets to be most effective, though likely they'll need significant maneuvering capability in order to meet their mission needs. Therefore, the ability to recapture such assets and service them should be valuable, potentially allowing refueling or recharge or on-orbit replacement of the prop module in its entirety. Also, we see the potential for benefit in the servicing of low Earth orbit constellations. A number of these are in development to meet global communications needs, and these typically contain a large number of satellites, which has resulted in a considerable amount of discussion around the topic of end-of-life disposal in order to avoid a proliferation of space debris. Here again, the ability to capture something small and possibly uncontrolled would be beneficial to enable active debris removal services. Now I'd like to spend some time describing our proposed demonstration. The core novelty of this demonstration is a new free-flyer capture system and technique, which uses a magnetic capture device to attach to a large, flat target surface on the free flyer. This greatly increases the chance of a successful capture by opening up the capture envelope, but it also creates an ambiguity in the spatial relationship between the robot tip and captured satellite. Our idea is that this ambiguity can be managed until the captured satellite can be berthed in a well-defined interface, from which point servicing may begin. To this end, the demonstration will proceed as follows. First, the robot will attach to the cube using its magnetic capture device while the cube is still fixed in its berthing port. The berthing port then releases the cube and the robot maneuvers it to a predetermined position. At this point, the robot releases the cube and we begin tracking it with the visual guidance system to measure its departure velocity. This shows our ability to perform a low impulse release with the magnetic capture device. When the operator is ready, the robot is commanded to approach and capture the now free-flying cube under the guidance of the visual guidance system. Incidentally, throughout this demonstration, the cube remains attached to structure with a retractable tether, so there is no risk of it becoming space debris. Once capture is successful, the robot is used to maneuver the cube back into its berthing port, and the berthing mechanism is actuated to hold it in place. This proves our ability to manage the ambiguous hold until an unambiguous berthing can be achieved, since we won't know in advance exactly where on the cube the robot is going to connect. Through this demonstration, we intend to raise the TRL of this capture system to 7. We currently assess the TRL of the major components at 4. The robotic arm leverages a series of prior developments at a smaller scale, and a limited capability prototype of this system has recently been demonstrated in the lab. The magnetic capture device has been developed up to the level of an engineering model and its core functions demonstrated in the lab. Environmental testing is planned to raise its TRL to 6. The VGS components, including target patterns and tracking algorithms, will leverage commercial or open source development and be adapted to our particular use case. Finally, I'd like to point out that this is a demonstration that can only be done on orbit, due to the great difficulty of accurately simulating free flyer contact dynamics on the ground, particularly in a case where the free flyer has a very small mass or an inertia. Also, that the ISS offers a unique and capable host platform for this purpose. Therefore, we've made some effort to configure the demonstration hardware such that it's able to fit within the Fram and Bartholomew payload envelopes for launch, and designed it to be optionally equipped with a Gold 2 connector for potential hosting on the Bishop airlock. With that, I thank you all for listening. Hope that you found this interesting, and that you'll feel free to contact me if you have any comments or questions. I look forward to hearing from you. Thank you.
All right, Atif, there you are. Go ahead and unmute. Tremendous.
Can you guys hear me?
Yes, we do. We
Great.
have
All right.
questions already.
Okay.
So you don't have to put up with me. So,
awesome.
First, is this an autonomous system or operated by humans?
I think the answer. Sorry for a cop out, but the answer is somewhere on this continuum, right? There's a part of this that's autonomous, but that doesn't mean that A lot of the operations aren't planned by humans and can't have human in the loop for safety or whatever, mission control. The capture part definitely has to be autonomous. But the way I imagine it, once the capture is complete and you have the cube in your hand and you're not really sure how you came down on the cube, then you have to update your situational awareness, your system model, and do a little bit of planning, and that can have human in the loop, or it can be autonomous too. There's algorithms that do path planning quite well. So the short answer is, yeah, it can go both ways.
Yeah.
Some part
Yes.
of it has to be autonomous for sure.
Answer is yes, both sides.
Mm-hmm.
So is there a prospective date for launch of this device?
There is not. It could be, there is a development path towards it. And so we have a sense of how, in what kind of time frame it could be ready for launch, but there is no current launch date plan for the demonstration here. We're proposing something. We want to know if there's interest in it. We think there is. We think there are use cases. We're interested in hearing from people, and yeah, that's all I'll say about that.
Okay. And a magnetic capture device might have some magnetic interference with the item captured. How do you mitigate any of the effects of the magnetic capture device?
So that's definitely something to think about. I will say the way the fields are oriented when the magnet is active, it kind of limits the scope of where they impact. So there's like a configuration solution to this problem, but definitely something that we're looking into. We have, like I said, we have a development path forward, and one of the questions we have to address during that path are EMI, EMC related questions, and I think the magnetic fields will kind of fall into that big bucket.
And well, we had somebody talking a little earlier about coatings that reduces charge development. So go through the posters that were before. Tell your daughter hello.
Yeah, sure, of course. Yeah, no, I see him. That's my son, actually. Sorry, can you repeat the question? I got distracted as they walked in.
I just pointed out that we had a poster about coatings.
I did see that one, passive control of spacecraft charging. Yeah, for sure. Yeah.
We have a question about space junk. Do you have any concern that the magnetic system could attract space junk that you're not trying to capture?
I don't think that the magnetic system has the reach for that, frankly. So, when you look at the distribution of space debris, it's, it looks bad on all the graphics that you'll see on the internet, but it's spatially not very dense. So the reach of the magnet is somewhat limited. I expect it to be, useful in the millimeters, centimeters, inches zone where I don't foresee that being a problem.
Okay. So there are lots of people that are developing systems to maneuver around, go and capture things. What is different about yours?
Yeah, no, totally fair question. I think a couple of things. One, that we are focused with this on the idea of capturing something that's uncontrolled, and that's a problem that people have looked into in the past. And I know that there are missions in development now to try to work that out. But we think this is a unique approach in that our parallel focus is also on keeping the economics in check. You don't want to end up with a system that is super complex that nobody can afford or only works in a very small niche kind of zone. We think this is a good broadly applicable system. The ways in which we have tried to keep it economical, we think don't affect its usefulness. And I think it still serves the purpose, which is to capture something that is not under its own active control, and maybe it's tumbling a little bit worse than you would want for your typical free fire capture operation. And also, we have in mind to keep the burden on the client as low as possible. So with this kind of capture system, you don't need a very complex fixture on the client. You don't need anything active on the client. All you need is a plate, frankly. And the targets are just stickers or paint or whatever. So that, I think that's what makes this unique, is we're focused on not just the functionality, which is new, but also the economics of it.
Okay, well, thank you for bringing it to us. This is a very comprehensive presentation and very understandable. And I appreciate you waiting until the end of the day.
And thank you for the questions, and thanks anyone who stuck around to listen. I appreciate you guys hosting me here.
We still have 50 people listening, so
wow,
you got a good audience.
All right.
So
good news.
This concludes our poster sessions for the 2020 ISS&D conference. And thank you to all of our authors and presenters, and to all who joined us on this segment. It's been a long segment. Thanks to Patrick and Sarah, who went in and handled all of our poster management and our author management as we went through this process. Now we'll take a break until 2300 Universal Time, or 1900, that is 7 p.m. Eastern Time, New York Time, 7 p.m. New York Time, 2300 UTC, where we'll get back into our technical sessions. And our first session is to look at JAXA capabilities and results. Thank you. Enjoy your dinner break.